Good morning. Welcome everybody on the second day. I'm happy to have you here, and just a kind reminder for all of you. So this event, I'm happy to have you here, first of all, but this event is about networking. So please enjoy the coffee break, enjoy the breaks, enjoy the aftermath of the conference, and find your new partners or old partners. Use this opportunity to meet again. And I'm also happy to lead both of the sessions today. And since my wife is still in Italy and giving this keynote about Aedes coraicus, I will be my wife now, so I will present the mosquito lab story for you. So the first presentation is like, it's kind of our vision now, and this is already happening, so luckily it's not just a vision anymore, but still it's quite in alignment with our ideas about the research on vector-borne diseases and what we decided more than 10 years ago here, and where we wanted to, I don't say end up, but at least which point we wanted to reach. And this is definitely one milestone we wanted to reach, to establish an ACL3 laboratory in our institute. And then if you are not familiar with the term, the ACL3 is arthropod containment level 3. It's like an equal to biosafety level 3, with the difference of containing the animals, the infected animals, in this case mosquitoes. So this is a very special lab space where you can literally infect mosquitoes and check on different parameters, basically to solve the most important question about mosquitoes: do they transmit different diseases under different conditions? And then this lab is like a simulation lab with proper biosafety standards. So it's a very special space. But to give you the idea about our history with mosquitoes, I think this group of animals, we work with them since 2009, and I can tell you that this is my favorite topic still. So most of you just know me as a Batman, but I was more a mosquito man before becoming a Batman, but not joking. So we started to work with mosquitoes in 2009, and we did the most important things which can build up a complex research strategy. So what is this? You start to do the surveillance, because you must know what's around. So we started to work with West Nile virus. We worked with West Nile virus not just in Hungary, but in the region. So we had some projects in Serbia. Now we had a project with our Croatian friends. So we are doing the basic understanding of the circulating strains of West Nile virus. And then when we moved to this building in 2012, we started to open new chapters in the research, and then we started to work with not just virologists, but also we had ecologists on board this time, so we were able to open to the ecology side of the mosquitoes, which is very important because this topic is super complex. So we started to work with invasive species. And then, I don't know if I can say, but we were, yeah, it's a bit funny, but we were lucky enough to have the Aedes coraicus here in Pécs for the first time in Hungary. So we had this role to work with this mosquito, and also we had, this is not a surprise, the Aedes albopictus 2014, 2016. So we started to do the specific monitoring of invasive species. And then step by step we built up international networks. We joined the Mosquito Alert application, citizen science project, which is a great project still, and it's still ongoing. I really suggest you to download the app, it's cool. And you can give us very precious data. We started to support the mosquito control activities in Hungary. I don't know how familiar you are with the Hungarian strategy, so it's basically 99% relying on chemicals. So we started to change this, first locally and then on the country level, by giving professional advice to cities, different companies, how to do properly the BTI treatment, the biological control of mosquitoes. And all these things are just somehow connected. So the basic question with mosquitoes for a virologist is like, okay, can this mosquito transmit this virus in Pécs, for example? But overall, the solution is like more complex. So you need to deal with the ecology, the control side, and everything. So this is how we step by step build this up. And then since the starting of the National Laboratory of Virology and since the establishment of the specific research group for mosquitoes and disease ecology, we started to broaden this research scope, and we started to do One Health-related research. So we started to speak with local doctors, try to solve questions like, why do we have increasing number of Virofilaria human cases in Pécs? We started to look behind this thing on the mosquito level, on the environmental level, on the ecological level. And then finally, we were lucky enough to win this grant. It's an Interreg grant, which we can use now to establish the lab. But before this, we were preparing. So we established a colony lab, where we started to practice how to maintain properly a colony. Also, we designed a digital dashboard to track all the parameters of these colonies, so you can repeat all kind of experiments. And now we have a stable colony of Aedes albopictus, which is a good start, but we won't stop here. So the next goal is starting and hopefully finishing the core of the laboratory this year. So why do we need this development? I don't know how familiar you are with vector-borne diseases. The problem with vector-borne diseases, if you hear something about, I don't know, an example, I have friends there, Valencia, okay. They publish a very nice paper that Valencian Aedes albopictus, the tiger mosquito, can and do transmit West Nile virus. Okay, so can you tell that this can happen in Hungary? Absolutely no, because all the factors from genetics to environment to the viral genetics and a lot of other factors which we just don't know, they are affecting the vector capacity of these mosquitoes. So the answer is you must test it locally, always locally, with your local population, local environment, local factors. So this is why there is an increasing number of these kind of labs and this kind of research done in Europe, because now this is the scientific consensus that the answer is not that easy. It's always some kind of local answer or semi-local. Let's call it regional. So we need in situ investigation. So we missed half of the mosquito team, and now they are here, but still missing some colleagues from the mosquito team because they were just visiting the Bernard Nocht Institute, and before that they visited other institutes to see the best practices, to see the best structure of these labs, to start the knowledge sharing and to start the networking part of this establishment. So this is a long way to go, but on the institutional level we are on track and we are building the networks. But why it's super important to build strong networks? This map gives you the answer. And if you remember the map of Biosafety 4 labs, you saw something very similar. So we already see our responsibility here. So after we establish this lab, we already do, and then we will have increasing responsibility for not just the Hungarian investigations, but for the whole region. So this is, again, just like the Biosafety 4, it's the last of these kind of laboratories in the whole region. So we really plan to do a lot of nice projects with you from Central Europe, Eastern Europe, Balkans, and beyond. So this is a unique opportunity for the whole region, and I can tell you proudly, and I'm happy to have a couple of the colleagues here, that we have a long track record, not just from Hungary, but from Serbia, Croatia, and other countries in the Balkans. And now, not just us in Pécs, but we together can step up with this kind of research. So I'm really happy to discuss the possibilities with you in the break, and then maybe find new questions to solve and new ways to go with this science. So this was the brief introduction. I hope I gave you some good ideas, and we can discuss any details if you want in the coffee break. Thank you very much. Okay, so now I will ask Dr. Johnny Ullman to present, no, sorry, I just missed it. Professor Daniel Ruzek, yes, to present about research on tick-borne encephalitis. Thank you.
Thank you so much, ladies and gentlemen, dear colleagues. First, let me thank to Gabor for inviting me. It's my great pleasure and honor to be here and celebrate this nice anniversary. Today I'm going to talk about my most favorite virus, which is tick-borne encephalitis virus, a virus which was firstly described in Europe in the former Czechoslovakia, in a country I'm from, in 1948 by Krejci, Rampas and Gallia. Unfortunately, Dr. Gallia got the infection in the laboratory and he died following tick-borne encephalitis in the age of 38 years. From that time, we have also the first descriptions of tick-borne encephalitis as a new disease. For example, in medical letters in 1949, there was a case report of a patient who was constantly repeating that Jesus Christ was the first socialist. So this is how tick-borne encephalitis can look like. Tick-borne encephalitis virus is a typical member of the genus Orthoflavivirus, family Flaviviridae, which is a group of about 70 viruses. Many of these are important human pathogens, such as West Nile virus, Japanese encephalitis virus, dengue virus, Zika virus, and so on. And tick-borne encephalitis virus is the most important representative of the tick-borne Orthoflavivirus group. Now we know that the virus occurs in large areas from Western Europe to Eastern Asia. Recently, the virus was identified also in the United Kingdom and North Africa. There's more than 10,000 clinical cases of tick-borne encephalitis reported every year, and we can see a high increase in the incidence of tick-borne encephalitis, especially in Europe, over the last few decades. For example, if we compare the situation here in these maps between 1995 and 2011, you can see a high increase in the areas where the virus can occur. This is mostly caused by global climatic changes. Ticks are moving to new areas, new endemic foci are established, and also ticks are moving to higher altitudes. Most people are infected with the virus during the feeding of an infected tick. But there's also another possibility. Some patients can get the infection after consuming unpasteurized milk or milk products, especially from goats, sheep, but also occasionally also from cows. The disease has typically two phases. The first phase is characterized with flu-like symptoms. Then there's an interphasic period, and the second phase is already characterized with neurological symptoms. It can manifest as meningitis, meningoencephalitis, or meningoencephalomyelitis as the most severe form. In Europe, the mortality following TBV infection is about 1% or less than 1%. But what's a bigger problem is so-called post-encephalitic syndrome, which is seen in about 25 to up to 50% of patients. And this is characterized with long-lasting or even permanent health problems after the acute phase. Here are data from our local hospitals, so you can see the mortality is less than 1%, but the post-encephalitic syndrome is seen in about 25% of patients and can be characterized as severe headache, tremor, sleeping disorders, vertigo, concentration problems, but also paresis or some psychiatric problems and so on. What are the problems associated with tick-borne encephalitis? First, it's the increase in TB incidence, and still the vaccination coverage in a number of endemic countries is very low. It can be delayed diagnosis or especially the limited ability to identify patients who are at a special risk of a more severe course of the disease. And most importantly, there's no specific treatment available for tick-borne encephalitis, and this is something we decided to change. In Russia and some other post-Soviet countries, there is available polyclonal immunoglobulin against tick-borne encephalitis. It's based on immunoglobulins that are concentrated from donors that were vaccinated against tick-borne encephalitis. According to the Russian sanitary rules, it's first important if you have a tick, you send it to the laboratory, and if it's found to be TBEV positive, then you can get these immunoglobulins ideally within the first 4 days after the tick attachment. And if it is given within these 4 days, you have almost 100% chance that the immunoglobulins will prevent you from development of tick-borne encephalitis. And if you get these immunoglobulins, you are protected for at least 1 month. In Europe, there were similar preparations available, Ensegam and FSME-Bulin, but in the second half of 1990s, these preparations were removed from the market because of the possible risk of antibody-dependent enhancement in some patients. Here we decided to develop a second generation of some immunoglobulins that could be used for post-exposure prophylaxis or early therapy of tick-borne encephalitis, now based on human monoclonal antibodies that are well characterized and where is no risk of antibody-dependent enhancement. We just started with a screening of our local patients for those who developed the strongest antibody response against tick-borne encephalitis. Then we invited these best patients to our lab, and they kindly donated half a liter of their blood each, and we isolated specific B cells from their blood and finally got a series of monoclonal antibodies that were tested further. So this is just a picture from the first screening. We had about 600 patients, and we identified these super neutralizators. Actually, this first screening was done by Charlie Rice in his laboratory. And then, as I said, we invited these patients, and they provided their blood. We isolated PBMCs from their blood and finally got specific B-cell clones. And finally, we obtained about 200 monoclonal antibodies that were screened against the virus in neutralization tests. And finally, we got these 4 finalists that neutralized the virus very effectively. These antibodies neutralized not only tick-borne encephalitis virus, but also other tick-borne orthoflaviviruses, such as Povassan virus, Chiasanur forest disease virus, Laupinkill virus, Langat virus, and Omsk hemorrhagic fever virus. And most importantly, these antibodies were effective also in the preclinical model of tick-borne encephalitis in lab mice that were infected with a lethal dose of the virus in pre-exposure design, and also there was good activity in the post-exposure design. We can see a nice activity even if the antibodies were given 3 days post-infection to the infected mice, which corresponds to the first phase of tick-borne encephalitis in humans. We also got the crystal structure of the virus, so of the antibody interacting with the virus, so we identified the epitope. And finally, we found out that a combination of 2 antibodies provides some synergic effect, is most effective, and most importantly, a combination of 2 antibodies in the cocktail provides a situation which is escape resistant. The virus is not able to mutate against these 2 antibodies at the same time, so there's no risk of mutations and escape of the virus under the treatment. So now these antibodies are under first phase of clinical trial in humans already, and hopefully one day these antibodies will be available for the post-exposure prophylaxis or treatment of TBE patients. But we can also target the virus using some direct-acting antivirus. Over the years, we have tested thousands of different molecules in our lab against tick-borne encephalitis, and we identified several molecules that are highly active against the virus. But you can imagine that the delivery of a new molecule to the clinics, to the patients, to the market is extremely difficult and extremely expensive. So the better way is to identify some molecules that are already approved for some other diseases or are under clinical trials for some other diseases, if these molecules have also some activity against tick-borne encephalitis. And indeed, here are some examples of molecules that exhibit some very good antiviral effect, such as remdesivir, which was approved for SARS-CoV-2 during the COVID-19 pandemics, or bemnifosbuvir, which is a compound under clinical trials against hepatitis C. So we observed quite nice antiviral activity of these molecules, and this was confirmed also in some advanced in vitro models, such as human brain organoids. Again, here you can see very nice inhibition of the viral replication, and also in brain slices, rat brain slices that are cultured ex vivo and infected with the virus. Again, very nice activity. The only little problem with remdesivir was some level of toxicity. But anyway, this compound is approved for the animal use, so the toxicity is not that big deal. So there are some compounds that could be potentially used off-label for the treatment of some patients with the severe course. But we can target not only the virus, we can target also the host to develop some antiviral response. Here we focused on CGAS STING pathway, which we stimulated with cyclic dinucleotide analogs. These compounds were developed by our collaborating chemists from the Institute of Organic Chemistry and Biochemistry in Prague, and we tested these compounds. We got also the crystal structure, and we tested these compounds and found that some of these are really very, very strongly potent. For example, the compound named MD1185 provides 100% survival in mice that were infected with the lethal dose of the virus. It works even at very low doses given. It works also in the post-exposure design. So this compound seems to be really very potent against the virus. Here are just some examples how the treatment can reduce the virus replication here in brain. So now we have some possibilities, at least experimentally, how we can treat tick-borne encephalitis. We can combine monoclonal antibodies, we can combine some direct-acting antivirus, and we can also stimulate the host immune system against the virus. Here I would like to just mention very briefly some other projects now focused on another tick-borne virus, CCHF. This is a very nice collaboration here with PAGE. We are trying to, it was also mentioned yesterday, we are trying to identify some molecules that are effective against this virus. In my lab, we use Hazara virus as a BSL-2 model for CCHF, and once we identify some molecules that are active against this virus, these molecules are then tested with the authentic CCHF here in PAGE, in BSL-4 labs. But our collaboration is based also on some staff exchanges between PAGE and Brno. So some students or postdocs came to here for BSL-4 trainings and doing also some research. And also people from PAGE came to my lab, such as Zoltán or Esther, doing some interesting experiments, and they were doing very well. So this is all from me. Congratulations again to the anniversary, and thank you so much for your attention.
Thank you, Daniel, for this great presentation. I really enjoyed, especially the pictures. Thank you for this. So the next one is Dr. Goran Vignievich, and I really like your title. So he will speak about our ongoing collaboration and shared goals. Thank you.
Yes. Good morning, everyone. Thank you, Gabor. I think in your presentation you already gave me a good introduction, because I will talk about today what we have achieved together and what we can achieve in the future. So when I grazed in a little bit of history, I realized that we have a collaboration between our biology department in Osijek and University in Page since 1970. It's not confirmed, but I know some of our professors have worked together, so it's like 50 years ago. I think that's when we started to actually move together in the right direction, and it was just a matter of time when we will cross our paths and start to collaborating more. I mean, also University of Osijek and University of Page are regional academic centers, so we have a lot of students, we have a lot of things that we share. Since we are relatively close to each other, both institutions are the parts of the same programs, same cooperations, and we are also committed to regional scientific advancement. So one of those programs is also Interreg framework, and as you mentioned in the first presentation, including this program, we have a new project that will work together. But prior to that, we also had exchange programs on regular meetings, scientific meetings, and we already share our ideas, methodologies, and discoveries. So yes, it is also, we have a good base to move on. And what we also achieved together, and it is, how to say, it's trendy now to have these collaboration agreements. Our both institutions agreed that we have, and we signed a cooperation agreement that will confirm our working together. So participation in scientific conferences, seminars like this, symposia, our exchange of experience, exchange of scientific and professional publication, exchange of students. So we have also this agreement that will confirm our cooperation. So if we move to the now spotlight, what we've done together, maybe prior to this project that we're working on together, we already have some publication, and last one was the detection of West Nile virus lineage 2 in our vectors in Croatia. With West Nile virus, it's tricky because what we found out is in Croatia we also have Croatian strain of, Hungarian strain of virus. So it's strain 2, and that is another confirmation that when you looking at the borders, it's in biological sense, no sense. So it is what we also confirmed that we need our cross-border activities to monitor and to focus on the things that we can deal together. So that's why we probably was successful to give this grant to have a Interreg project. It's a big project, something around 6 million euros, and this project will enhance our capacity to fight mosquitoes, to improve mosquito control, and with this project, new laboratory that you mentioned in the first presentation, will be here in this institute, and on our side, on Croatian side, there will be a new capacity, new building for mosquito control. It will be called Mosquito Control Center. These are some visuals, how will it be in future. It is now just started to make some progress. This is the first year of the project. So the Mosquito Lab project is focused to implement new biological control and to control mosquitoes in a different approach, integrative approach that will also include biological control. So this building will be also equipped with SIT laboratory, sterile techniques that will help us fight invasive mosquitoes, especially Aedes albopictus, Coraicus, maybe even Aegypti. So also we will try to move our focus as well to detecting pathogens in mosquitoes, also to fight resistance, insecticide resistance in mosquitoes. So we will try to induce this biological control more and more and to reduce chemical influx into our environment. So what we are achieving with this project, that we are strengthening disease surveillance, outbreak response, public health infrastructure across both nations. It will help protect our biodiversity, monitoring ecosystem, and we'll try to promote sustainable development. All of this, of course, is supporting EU goals in the future. So we have a good base, and we are trying to do more. Since we are closely connected, Peć and Osijek are, I think, about 100 kilometers away, so we are relatively close, and that gives us a lot of opportunity to exchange, to do things together. And we will try to do it in the future and to deepen our collaboration, expand research, and all the things that we can do to strengthen, to advance knowledge, health system, regional prosperity, and also transforming the mosquito control from these, how to say, traditional ways to more novel ones. And that's it. Thank you for your attention. For more questions about mosquito control, in a coffee break.
Thank you, Goran. So the next one is Dr. Pavle Banovic, and I see your title, so you will speak about another very important regional project. The floor is yours. Thank you.
Thank you, Gabor. Thank you for having me here. Thank you all for being here. So, how many times you have heard, who are you to implement something new? We are doing this thing for almost 20 years, now you want to change it? Or why should we start working with new pathogens? We already have enough job to do. We don't have equipment, we don't have money, we don't have people. It's the best if we can continue with working of the things that we are already doing for decades. Why should we change anything? How many times you have heard this? And then million obstacles. You don't have money, you don't have a people to talk with, you don't have a local support. You want to do something, you have an idea how to do something, or at least how it should look like, but basically you are alone on the shore. Do you have this feeling? Have you had it sometimes? And if you want to do it something, you need to do it by yourself. Because all others are smart enough to know, don't go in new directions, a lot of pitfalls, a lot of problems. God forbid you make a mistake, then you are stupid. And all people who told you that you should do anything new are right away smarter. Well, this is a story that is not done, not nearly done, but very interesting story of underdogs working together. To be clear, I am the underdog. Working together with the good people, making friends, making nice stories, nice results, changing the environment for the research for many other people, changing the environment of public health, of the research, and most important, communication pipelines. So, tick-borne diseases, vector-borne diseases, we heard a lot about it today and yesterday. We know that we have a lot of problems. We have new pathogens, we have new diseases come, we do not know that it is a new, we test it for everything, then it's unknown origin, then we wait to see what the bigger country will say, oh, it's pathogen this, it's pathogen that, or you send to Central Laboratory and you hope that you will get something from any type of positive finding. But we always talk, we always speak. We have a climate change, we have introduction of new pathogen, we have introduction of new vectors, we need to surveil, we need to monitor. Yes, but where is the money for that? Where are the people for that? Somebody needs to come outside and do this. Somebody needs to pay these people too. Okay, maybe they will do it for free, but not forever. But on the other hand, you have a lot of researchers that are eager to work and eager to do something, and they say, no, you cannot work with human samples. You are not a medical doctors. You are not a medical institution. You're only researcher, like only researcher. Why we are making this type of boundaries for different sectors? Nobody is stealing other job. We are just working together. And this is the story of working together. So we knew that the Balkans are diverse, let's say geographically diverse. And we knew that we have pathogens that are waiting to be not discovered, but registered and described. Because when you look at the register of the medical history, at least in the city where I work, in Novi Sad, or as you will say, Uvidek, you see that there is a lot of encephalitis of unknown origin. Patients are getting dismissed home, they are getting healthy, and they go home. Unknown origin, viral infection of unknown origin. And nobody asks why we have this type of infection and what is the cause in this type of infection. When you speak to clinician, they will say, okay, if I know it's tick-borne encephalitis, do I have a different treatment for this patient? No. Then why do I care? Is it important for me if it's a West Nile and tick-borne encephalitis? I say, yes, because you can prevent one with a vaccine. And they're, oh yeah, but we don't have information about it. And now we are going into a vicious cycle. We do not know that we have a pathogen. We do not know where it circulates. We may have a disease, but we cannot confirm because there is no clinical suspicion out of it. We needed help from the scientific team, from the research team, because healthcare institution cannot do groundbreaking science, at least not in Balkans, as two main factors: human resources and money. So, we all know what are the biggest problems. We have the mitigation of hosts, the animals that serve as for the blood meals, for the vectors. We have the movement of the reservoirs that can be mosquitoes in the case of mosquito-borne pathogens, or with the ticks. And we have expansion of the vectors. So, okay, we say they are going to the north, they are expanding, but once in a while we get some surprises. So last year we got cases of CCHF in Greece. I mean, nobody expected that. 2 years ago we had the cases of CCHF in North Macedonia. Nobody expected that. Nobody expects spreading on the south. Something is happening. So we need communication. We don't have tools and resources to do this by ourselves. The consequences, of course, we have new pathogen foci that are formed, and we have diseases that are not expected to appear in this type of, let's say, environment, this type of terrain. We have clinicians that are encountering new diseases, neuro-invasive diseases, or hemorrhagic syndromes that are not clear what are the let's say, caused by. The etiology is not clear. For hemorrhagic syndromes is the easiest because there is not a lot of pathogens that can cause that. But nevertheless, it is important to mitigate the spreading and to have the accident of exposure of the healthcare personnel working with the patient. And finally, we need to have laboratories that can detect endemic and introduced agents. We need to be prepared. When you work alone, you cannot be prepared. So, we made a Balkan model, and because it's ours, it's the best. So, we need to be proactive. We need to search for something. We need to surveil what is happening in the region. We are stepping out of the pipeline. We are communicating. We need to combine the human medicine, the vet medicine. We need vector ecology and basic research knowledge. Wearing the white coat in a hospital does not give you all knowledge the world has, not even ChatGPT. We need to talk. So, only in this way we can have joint investigation, and we need to have the shared analytics of the problem that we all share. There was a nice sentence on the previous presentation, the rivers that do not divide but connect. That's very good. So, we need to find a way that are connecting us, not dividing, especially because for some reason people think that it is a shame to ask. That is a problem to not know. No, it's a good thing if you identify there's something you do not know, so you are aware what you need to pursue. So, this approach with the communication pipeline allowed us to observe the clinical cases, to observe the changes in the field, to sample vectors in the field, to organize household visits. So imagine you have a patient that has rickettsial disease. You don't have a clue how this disease happened in this patient. You need to go to the household where this occurred. You call the vet, you call the biologist, and to go to household, everybody do its job. I'm talking with the family, vet is doing stuff with the animal, biologist is looking around in the environment, trying to see what is happening with the ticks. Finally, we found out, actually, long story short, the husband of our patient was actually a forester, working in the nearby forest. He went back home. He did not remove his jacket, his working suit at the office. He removed it at home, all ticks from from his jacket went to the let's say, nearby clothes. And of course, as a Murphy law will say, one of these ticks went to the clothes of his wife, who got infested by tick, who got a rickettsial disease. So the conclusion of that research is, of course, it's a husband's fault. And that was in alignment with the previous researchers, yes? So that was an easy pass on the reviewers. Joke aside, so this framework of Balkan Association for Vector-Borne Diseases allowed us to not be afraid to talk with the colleagues about the topics that we do not know. It is okay for me to know how the disease is manifesting. It is okay for me to know which organ will be affected. But ecology of the pathogen or behaving of the animal, ecology of the animals, that is not a thing that medical doctor can know. And that is a thing that is crucial to know if you want to prevent next cases. So, we wanted to uplift our work. We are National Reference Laboratory for rabies for more than 100 years, and that is something that is okay for us. We did different zoonotic diseases, and that is okay, that is very nice. But then, you had a problem such as West Nile virus, you have a problem such as tick-borne encephalitis virus. You expect to have it, you don't have any diagnosis. There is no laboratory that can give you final confirmation. As I'm aware, people from Serbia sent samples abroad to be tested for tick-borne encephalitis. Okay, this should not be a big problem, right? Well, not so right. You don't have knowledge to build BSL level 3, you don't have money to build BSL level 3, you don't have a clue where to put it, you don't know what to do with the pipes, you don't know what to do with electricity, you do not know what to do for the fail-safe technology, you don't know a thing except that you want to work with this pathogen, that you want to increase the level of preparedness, that you want to learn something new. And then there was cost action, Aedes invasive mosquitoes, and I applied for STSM, Gabor and Cornelia accepted me. We did the mosquito stuff, but we also did biosafety stuff. And that set me the ground to see how the laboratory should work. I knew that we cannot do classical tree, because especially in 2019-20, it requested to throw away money into this laboratory regardless of what do you need it. But with the help of colleagues from Hungary, we were able to identify the problems that are actually the biggest for us and where we should invest and how to modify our procedures. So they provided us with high containment expertise, the advanced molecular methods, training platforms, and they are our solid and one of the strongest partners in the region. Forever what we want to do, what we work, they are here for us. And also, if we can help in any way, we are more than welcome to, we are more than happy to help. So, how would it look like? So, of course, we need to do risk-based laboratory development steps, risk assessment of sampling, healthy storage of material. Actually, I just asked, what do you do with this type of samples? And the guy just told me, you do this, 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 this. Simple as that. Risk mitigation, planning, implementation of new analytic systems, Nanopore, fantastic tool. And staff training. The guys came to Novi Sad several times to train us. We came here several times to be trained. That is opportunity that was more valuable than gold for us. From the beginning, when I started this presentation, I told you, everything that was proposed was, who are you to change anything? Why would we do something else? You don't have money, you don't have people. Suddenly, you don't need a lot of money. Suddenly, you don't need so much people because you have colleagues that are able to help you and provide you the most simple way, most effective way, how to fulfill your task. And only for one reason, they are good people. So, several photos. I did my best to dedicate only photos from the laboratory. There will be no photos from celebrations, from the fieldworks, and the BAVBD annual meetings. For that, you need to go to the BAVBD website. You need to lift off the visits to the website, please, and share it. So, This is the last visit of Patch team to our institute. We have an interesting project. We opened like 15 bottles of old vaccines against rabies, and now from all around the world, from Siberia to New York and Canada, and Western Europe, and Eastern Europe, and Balkans. And now we are trying to see if we can recover some of the genetic material from it. So, what I was talking about for the previous, let's sum it up. So, initially, in my opinion at least, ideal input is clinical or field input. Okay, so we have something, we found something. Let's say we have the yellow lamp on. So we have ticks from humans, we can find something in ticks, we can find something in human blood, in household animals, or in the vectors that are in farms. Then we do a BCL2 screening, 2+ screening, so we can do PCR now, we can do serology, we ID ticks, and now we can do a nanopore. Oh, sorry, we will be able to do a nanopore, we only bought it. We need to be a little proficient to be able to do it routinely. Then we are able to trigger one health visit. Let's go back, let's call the colleagues and then examine holistically. And then the big guys step in. We ask for BSL-4 support. So isolation of the viruses, NGS, and biosafety monitoring. Now, if Gabor decides that something is good for any kind of animal model or anything else, they are the guys who know what to do next. I'm the one only can hypothesize and ask a question if it's possible. And finally, when we wrap it up, we are able to make public health action. We are able to change. We are able to save some life, even if it's one life. Even if it's a reduction of disease sequelae. So, as Professor Ružić says, when you have tick-borne encephalitis, sometimes you die, sometimes you survive, especially in East, but you get paralyzed, you have problems. So even if you prevent one of these cases, and this guy do not have encephalitis, and this guy do not have a paralysis for the rest of his life, we did something. It's not so big, we don't need to have mass-scale changes. We can, depends what we are fighting against with. So the core methods that we use here are actually PCR screening, morphological identification, serology and confirmatory approach, sequencing, and integration of biosafety and biosecurity measures, and finally, risk communication pathways. So we use tick-borne encephalitis as a model for this because for us it was the easiest. There is vaccine available. The virus is, we assume this was circulating in Serbia. There was not a lot of direct evidence, only 1-2 detection in ticks in whole country. But our plans were a little bit changed when we found out that there is a CCHF outbreak in North Macedonia. So we were like, okay, move tick-borne encephalitis for a side, let's do this. And we did a really good job. Nobody died, first thing, in any country, I mean, of the team. We successfully identified the viruses, lineages. We checked the healthcare workers for their exposure. We checked the animals on the farm where the outbreak happened. We did everything so rapidly that the teams from other countries were a bit surprised. But that's why the Balkan model is the best model. So, the results. Yes. Yeah, we like to go to the news also. So this is our vaccine project. I mean, we will not prevent pandemic, but it sounds good, right? And yeah, we went to United States also. This is we like to work with biosafety. Balkan Association have a new task force for biosafety, and Balázs and his team are in charge of it and very, very, very serious about it. The picture is not serious, but we are boys. So for everyone who is interested in biosafety work or tools or anything similar, I would invite you to, again, check the website, and if you want, you can join to the task force. Balázs is very active. He is planning to initiate some webinars, some meetings, and I think it will be very good for everyone. And the best thing, everything is for free. So, outcomes. We now have enhanced diagnostics. We can diagnose tick-borne encephalitis virus without a problem. Serologically, we implement a neutralization assay. We have colleagues now very close to us. They can do isolation neutralization assay for CCHF. Very good for us. We are expecting epidemic in South Serbia, maybe this year, maybe next year. Nobody's talking about that. We have colleagues that we can go and say, we need more training. We are not prepared for this problem. Can you help us? And they always welcome us to new trainings or even come to our lab and help us. And together we are prepared for events, not only Peć and Novi Sad, all of us. I assume that we can communicate casually, let's say, and without any constraints of any bad intentions. There is not a lot of us. And yesterday there was a really good slide. There is less BSL-4 laboratories than astronauts. But also there is not a lot of scientists and people from the, let's say, clinical part of the world that are willing to communicate together. This is, I think, our biggest strength, and this is something that we need to work on, the communication and the sharing of the information, because something that is very complicated, something that is unreachable for me may be a routine job for you, and vice versa. Maybe you are desperate for some samples, maybe you are desperate for some diagnostics that is unavailable in your laboratory, but for me it's a routine job, and that is something that we can gain the biggest momentum. So our priorities, we need to scale up, we need to make Balkans great again. We need to standardize SOP. We want to unify our work, because ideally work in one lab should be analogs to the work in other lab. We want to expand the confirmatory serological assay, and that is something that the Gabor team is working a lot on, but that is Sentinel project, and there you will have probably more information later, or you already talked about it. I'm sorry if I missed. I'm sorry, Gabor. And we want to deepen our knowledge on vector ecology, and we want to deepen our knowledge on vector-borne diseases, and this is the only way that we can work together and to join and to address this together, in a line together, and to defend each other in the scientific way that some measures are necessary, even if they are expensive for the governments, even if they are expensive for the public health. It's always is more cheaper to prevent something than to treat it. That is the golden sentence that we don't forget, but the guys that have decisions are sometimes not remembering that. So I would like to finish this presentation. I'm sorry if this was a detour from the scientific presentation that you had earlier, but I think that it's very important for us to have in mind that we have a chance to change something. We have a chance to uplift ourselves, our colleagues, and it is the only factor that is deciding if this is happening or not going to happen is us. So please take in consideration advice from Gabor. This is an event for networking, and if any one of you would like to or have any idea what we can do together, please let me know. I'll be more than happy to make some plans today. Thank you.
Thank you, Pavle, for presenting our shared vision. So the next presenter is Professor Florian Kramer. I was happy to see that there will be a topic on hantaviruses, so please, floor is yours.
All right. Okay, that's Hungarian. First of all, thanks for inviting me to come here. This is super interesting for me to learn what's going on in this region. My lab is typically very interested in influenza viruses, but I decided to talk about our work on hantaviruses today. So a few years ago, we started to work on Andes viruses, and then I got interested in Pumala virus. This work started in my laboratory in New York, and we are now transferring a lot of this work to my new lab in Vienna at the Medical University of Vienna. So just a little bit of a background about hantaviruses. Pumalavirus is a hantavirus, an orthohantavirus. These viruses are relatively simple. Here on one side you can see the virion structure. We have 3 genomic segments, and then we have basically glycoprotein on the outside. We don't even have a matrix protein for these viruses. These 3 segments code for a polymerase on the large segment. The M segment codes for a glycoprotein, which is very interesting, and I'll talk a little bit more about that. And then the S or small segment encodes for a nucleoprotein and a non-structural protein. As I mentioned, the glycoprotein is interesting. It's a relatively complex glycoprotein. I'm coming from the influenza world where we have 2 glycoproteins, but they are relatively simple, easy to express, and have been characterized very early on in terms of structure. With hantaviruses, this was very complicated, and only in the last few years we have learned more about the structure of the glycoprotein. It's basically divided. It has a GN part and the GC part, and as you can see here in this, I don't know if you can see my cursor, probably not. On the right side, you basically have 3 transmembrane domains, which is unusual, at least for an influenza person, when dealing with glycoproteins. And basically, these GN and GC parts come together and then form a dimer, and that dimer forms a tetramer that you then can find on the surface of the virus. These glycoproteins are extremely hard to express. This is actually a challenge for vaccine development, and it's also a challenge for structural biology. Specifically, I'm focusing on Pumalavirus. Pumalavirus was discovered in Finland originally. And it's carried by these bank walls, these rodents that have spread throughout Europe, but also into parts of Asia. And typically, these rodents carry the virus. They're asymptomatic. They don't have symptoms, but they excrete the virus via their feces and via their urine, and then people can get infected. Pumalavirus infections are actually some of the milder hantavirus infections, and typically the disease is hantavirus fever with renal syndrome, but the milder version that's called nephropathia endemica. A little bit about the infections. Typically people get infected when they inhale these dried feces or dried urine from these animals. That often happens when people clean their garage, for example. You have feces there. People kind of basically use a broom to clean the garage, and then dust is aerosolized, and people inhale that. And then you have a relatively long incubation time, 2 to 6 weeks, and then initially you have relatively aspecific symptoms, flu-like symptoms in a febrile phase. And then followed by that, you often see kidney damage, and then most people recover. So compared to New World hantavirus infections, where the case fatality rates are often around 50%, the case fatality rate here is only about 0.5%, but very often these patients have to be hospitalized, and the disease can actually be pretty severe. I'm interested in that because, of course, there's a lot of cases in Europe. These are numbers from 2019. And you can see these are absolute numbers, so this is not normalized per population. But you can see that there are some countries that actually have a relatively large number of cases. That includes Finland, but also Austria, Sweden, Slovenia. And I'm looking at this, of course, from the Austrian angle. And just to show you a distribution of these infections in Austria, this is a historic map where you can see that specifically the south of Austria is affected. So the state that has most of the infections is Styria, but there's also infections in Carinthia and in Burgenland. And of course, if you go south to Slovenia, you have a lot of infections in that same kind of region bordering Austria too. And so a few years ago, we started a cohort study with 2 physicians from Graz, which is in the middle of that endemic region. We worked with Robert Krause and Stefan Hatzel to recruit patients and follow them longitudinally through the course of infection. So they get diagnosed, they get hospitalized, and then typically we get a sample from them right away when they are hospitalized. So this is this T1. It's typically about 3 days after symptom onset. Then we take a sample a little bit later, about 7 days into the disease. Then one about 3 to 4 weeks later, and then one in the convalescent phase. And we were really interested in how the immune response to these infections develop. And we were mostly focusing on immune responses to the glycoproteins. And that's something that hasn't been studied very well, not in a longitudinal fashion for Pumala virus. We have a lot of data. I don't have much time, so I'm just going to show you some of the data that we generated. And I'll start with neutralization. Pumala virus doesn't grow super well, and it's actually not so easy to do neutralization assays in a high throughput fashion. So what we do here is we use VSV that is pseudotyped with the GNGC protein of Pumala virus. This was done in a collaboration with Kartik Chandran at Einstein University, who has made these viruses. And so we use them in a biosafety level 2 laboratory, and we can do relatively large numbers of neutralization assays with that. And what you can see here is basically neutralizing antibody titers throughout the course of infection. And you can see that at the time point 1, so when people are hospitalized, they already have neutralizing antibodies in their serum, and these neutralizing antibody levels go up over time. And on the right side, you see a negative and a positive control. So that is expected, right? You get the infection, you recover, you get more and more neutralizing antibodies, and the immune system responds. I'm coming, as I said, from the influenza side, and I've been working for many years on cross-reactive and cross-neutralizing antibody responses, and so we were super interested in seeing how cross-neutralizing this sera would be for other hantaviruses. And so we also ran neutralization assays against other Old World and New World hantaviruses. And what you can see here is representative figures from some of these strains. So in B, you see neutralization of Sol virus, which is a related Old World hantavirus, but it's not that closely related to Pumala virus. And what you can see is that a proportion of these infected individuals actually have neutralizing antibodies also against Sol virus. And specifically, I wanted to draw your attention to time point 4, when you have an increase in the number of individuals who have these cross-neutralizing antibodies, but also in the titers. The titers get higher. We also looked at Dobrava-Belgrad virus, which is another Old World hantavirus, which is actually prevalent in the Balkans, and we basically see the same pattern. We see some neutralization from the beginning in some of the patients, but over time, we see more and more patients developing these neutralizing antibody titers, and the titers go up. And interestingly, that even crosses over to New World hantaviruses. So on the very right side, you see neutralizing antibody titers to Andes virus, where we see exactly the same pattern. Some patients have neutralizing antibodies from the get-go, but at the last time point, the titers are higher, and it's a larger proportion of individuals who have these antibodies. So it seems that there is a lot of affinity maturation going on and the broadening of the immune response over time. We also looked at the different subtypes of IgG. We also looked at IgA and IgM, but I don't have time to show you that, over the course of the infection. And here we see something very interesting. On the left side, you see IgG1 titers, and these IgG1 titers go up over time, and that's not unexpected, even though you would think that between T3 and T4, there's not much of an increase anymore. In an influenza infection, you would actually already see a little bit of a decline there. We still see an increase. As expected, there's not much going on with IgG2 and IgG4. Those are not subtypes of antibodies that you would expect against glycoproteins of viruses, even though IgG4 can sometimes show up for SARS-CoV-2, for example, or Ebola. But what we also see is an initial IgG3 response, and then a decline of the IgG3 response. and some of the IgG4 is coming up at that latest time point. If you look, IgG3 is going down, and some IgG4 is coming up. And I mentioned Ebola earlier, and there it has been shown that you can have IgG4 responses very late in the infection and when people recover, and that is potentially associated a little bit with persistent presence of antigen. And these changes in subtypes actually have functional consequences. So you can look at virus neutralization, and over time we see that that goes up. But you can also look at FC-FCR interactions for these antibodies, right? Antibodies not just neutralize viruses, but they also engage with effector cells like macrophages and NK cells and recruit them to infected cells, and that's done by FC-FCR interactions. And what we see is that in the last time point with this decline of IgG3, we also see a decline in FC-FCR interactions. And that makes sense because IgG3 has a very strong interaction with FC receptors, but IgG4, for example, doesn't interact very well with FC receptors. So we think that can have functional consequences over time, but it also tells us that there are things happening very late in the infection or in the convalescent phase. And we actually think that there might be a continuous antigen present over months in these patients, similar to other virus infections where that has been described. Just switching topics a little bit here, we saw these cross-reactive antibody responses, and we were super interested to see if we could also isolate monoclonal antibodies with these traits. And so we worked with Patrick Wilson at Cornell University, and we actually took PBMCs from some of these patients, and we sorted for B cells, in this case memory B cells and plasmablasts, and tried to clone out monoclonal antibodies in the hopes of identifying monoclonals that could cross-neutralize a large number of different hantavirus strains. And we were lucky. On the left side, you can find a panel of antibodies that show relatively broad binding to different GN-GC proteins of different hantaviruses. Specifically, 3 of these antibodies stand out because they bind to all the tested GN-GC proteins. And on the right side, you can see the neutralizers. And we have at least 2 antibodies that seem to cross-neutralize almost all of the tested hantaviruses, including old world and new world hantaviruses. And we're now in the process of characterizing these antibodies better and actually trying to develop them into potential therapeutics. And we have started to work with Pablo Guardado Calvo at Institut Pasteur in Paris, who has solved some of the first structures of these GN-GC proteins. And he also looked at the structure, the complex of one of these antibodies, antibody 1295, with the GN-GC. And we have now characterized the epitope that this antibody binds to. The remarkable part here is that it's actually a GN binder. So GC is more conserved than GN for hantaviruses, but this cross-neutralizing antibody actually binds to GN, potentially a new cross-reactive epitope. So to conclude, Pumala infection induces strong homologous binding and functional antibody responses in humans, but we also see heterologous responses to divergent old world and new world hantavirus GN-GCs. And these heterologous responses increase over time. And we also have this very late switch from from IgG3 to IgG4. And we think this could be potentially a hint that there is persistent antigen present, maybe in immunoprivileged sites, or maybe even infectious virus. So I find that very interesting, and we're now doing follow-up studies in Austria to see if we can detect virus in some of these patients months or even years after their initial infection. And so some of these monoclonal antibodies to GN-GC may be future treatment options for infections. And so I just wanted to thank the team. Most of this work is done by a very talented assistant professor in my lab, Jordan Clark. And as I mentioned, we have done this together with the Medical University of Graz, with Robert Krause and Stefan Hatzel, and then of course with Pablo and Patrick for structural work and the monoclonal antibodies. I just wanted to mention that we are very interested in collaborating specifically with clinicians. We would like to look at other hantavirus infections. We have compared this data to Andes virus infection cohorts in Chile, and the patterns are different. You don't see this IgG3, IgG4 switch there. So it seems that New World hantaviruses, specifically Andes, induces antibody responses that look different from what Bumala does. And I would be very interested in looking into the Brava-Belgrade cases, for example, or Sarima infection cases, to compare and see how that looks like. And the other point that I wanted to make is we're very interested in monoclonal antibodies, not just to hantaviruses, but also other vector-borne viruses, for example. And we have now access in Vienna to a mouse model that actually expresses human IgG germlines. And so we can make in mouse models human monoclonal antibodies. So if anybody's interested in collaborating, just talk to me. Thanks.
Thank you, Florian. The next presenter is Professor Kurt Zatlukal. I think this is really a hot topic now. So he will speak about the concepts for rapid response to the next pandemic. Please.
Thank you so much. Yeah, it's my great pleasure also to being here and to share a little bit also some of the work we are doing together here with the team in Page. And when we talk about pandemic preparedness, this has gained a major international relevance. And I think the level of relevance is most likely underlined if there is a budget committed. You know, paper is very flexible, but if there's really the willing to spend, to invest money, I think that's a strong evidence. And for instance, there was the Moonshot program launched under the Biden administration with $88 billion on biodefense. And even on the G20, they committed $10 billion per year for pandemic preparedness. And the major goal is to be able to more rapidly respond to ever what the next big threat will be. And this is materialized also in several so-called 100 days missions. There's just two examples, one published by the US Academy of Medical Sciences, the other from CEPI. Now, what is behind these 100 days missions? It's really to make available diagnostics, vaccines, and medicines within 100 days of an emerging new threat. And we are now considering what technologies are needed and have to be aligned to make this possible. And so I will give you a little bit a short insight into the various project activities we have in this direction. So actually it starts with identifying the pathogen. And they're very important, you need a sequence. Without the sequence, you cannot start your programs. So therefore we think about how we can more rapidly access the sequences of a pathogen. But then the next thing is we need technologies that allow to rapidly produce diagnostics, vaccines, and medicines. And here we are using a specific plant-based manufacturing technology. Now, showing you a little bit more detail, core element is, of course, our laboratory we have in Graz at the Medical University. And that's not a classical BSL-3 lab because it was specifically designed to do autopsies under high containment conditions because we are part of a pathology institute. And if you do pathology in a high containment environment, you're exposed to higher risk because you take sharps, you get contaminated, so you have to counterbalance the increased risk by increased biosafety. So that's why we actually have implemented certain biosafety measures like a chemical shower you otherwise would only find in BSL-4 lab. And here we're very glad to the training we have got here, how to really work under BSL-4 condition, which lays the foundation considering upgrade to a full BSL-4 lab, which is under discussion now. Now, giving you some example why autopsy is an interesting element in the context of this overall rapid responses. For instance, these are one of the very early slides, how a COVID, SARS-CoV-2 infection damages the human lung. You on the one hand see that there is a barrier preventing the oxygen transition. You can see which cells are infected, like the alveocytes, and you see also the role of comorbidities. But even more, you can get molecular insight. For instance, you can do transcriptome sequencing, even in case of autopsies. And on the left, you see on the one hand, patients who died without COVID, the others that had COVID. And if you look closer, COVID is not a homogeneous disease. There are different mechanisms driving the disease. And depending on which mechanism drives the disease, you need different medicines to prevent it. And also, if you design a clinical trial, you have to consider whether you have to stratify the treatments. And actually, on infectious diseases, we need targeted, personalized medicine like we have in oncology. Another element is quite interesting on the sequencing side. So we have some initiatives now running into making field sequencing more accessible. And on the left, that's an example of a technology developed in Japan. Actually, it's a spin-off of the Riken Institute. And actually, what you see here, this is a single-use device, a cartridge where you just put in the swab. This does the whole sample processing, and at the bottom, you have a battery-operated detection device. So you get an amplification reaction, and you have the results in 20 minutes. And this technology is now further developed to do the sample processing for sequencing, for instance, nanopore sequencing, and on the other hand, we're working on solution doing the bioinformatics analysis in an equipment that is suitable for field use. Now, considering what approaches are needed to making medicines more rapidly available, and there are different strategies to do it. On the one hand, there is a major interest developing drugs that have a wide spectrum, so that you have inhibitors that can be used for various families. Antibodies and vaccines with wide cross-reactivity, and Florian Kramer, I think, nicely showed you what approaches could lead to these solutions. But also drug repurposing using computational modeling play here a major role. And then you need rapid and flexible scalable manufacturing technologies, and also clinical approval procedures that allow much faster market authorization than we have in classical drug development. Now, some examples. For instance, we participated in some programs where they did computer-based simulations. For instance, here they screened 1 billion compounds that could prevent SARS-CoV-2 replication, and we tested them. And maybe you remember the presentation from yesterday, how these type of experiments scale up immediately. You have to have different concentration in different cell models, in different virus lineages, so you need significant laboratory capacities to complement computational modeling and to really find the relevant compounds. In this context, we actually found out that the concept was the most active one, much more than the chemical compounds. It was using a biologic, using the whole receptor, the ACE2, to block the virus from infection. The interesting part is when you use a complete receptor as a neutralizing agent is that essentially you avoid immune escape. And this you see here, we compared, for instance, on the left, the ACE2 decoy. You see actually its neutralizing activity even increased with the emerging variants. Yeah? So we could about 4 times had higher neutralizing activity with the latest Omicron variants as compared to the original Wuhan. In contrast, if we take sera from convalescent patients, you see a marked decrease. Patients who got the immune response induced in the first wave, they had about 70 times reduced neutralizing activity in the Omicron variants. Even more pronounced if you have a monoclonal antibody that recognizes a narrow epitope. On the other hand, some chemical compounds like the AMPRO inhibitor from Pfizer stayed more or less constant for the various variants. Now, these decoys are also very active in vivo. So we did a therapeutic hamster study. Actually, mice received the virus intranasally, and on the next day were treated with the decoy. And you can see on the one hand, this reduced weight loss, and on the other hand, also markedly reduced the virus load in the lung. And this was quite challenging because it was a therapeutic approach, so they were first infected, and we started with the decoy on the next day. A very interesting observation we made, when we produced this decoy in plants instead of classical biomanufacturing in CHO cells, we did not only see that, of course, you have neutralizing, but even the neutralizing activity was increased. And in this case, we found up to 100-fold increased biological activity. And one possible explanation is for this is the different glycosylation you have in plants, and here we work using glyco-engineered plants. So controlling glycosylation is a very important element in developing antiviral agents. Now, actually, with this observation, we got major funding now from the European Defence Fund to make this technology available in context of rapid responses and to bring it from the laboratory to an industrial scale. And why is this that interesting? Think about the whole complexity of classical biomanufacturing is replaced by a plant, because biology is doing all the job, the quality control, which otherwise needs very complex engineering resources and technology. And this, of course, is of great interest, having a robust platform available that even works under the threat of a pandemic or other crisis. Another advantage is the rapid production. And actually, this is possible because you're using a transient expression system. So you infect the leaves of the plants with agrobacteria, which is cloning agrobacteria is a very simple process. You infect them naturally, and within 5 days you can harvest the protein from the leaves using classical purification downstream technologies. And the next advantage is, once you have your positive result in the single plant, you can easily scale up to industrial scale just by growing more plants. So you can imagine this scaling up is much easier than in classical production. And on the right, you see an example from Medicago in Canada that produced a COVID vaccine. Now, the next bottleneck is the regulatory path. You know, classical drug development may take up to 15 years. And in COVID, I think it was a really masterpiece that they were able getting the vaccines ready for approval within less than a year. This was possible because it was not a sequential approval process, but it was a parallel process, but also following special approval procedures. And actually, there are two different ways in cases of some emergencies. For instance, in the US, there is the animal rule, so that you can replace the efficacy studies in humans by doing animal studies, extended animal studies. And we have a very similar regulatory path in Europe, which is called exceptional circumstances by the European Medicines Agency. So you're shifting the work from the clinical studies to extended animal studies. And in this context, we are exploring alternatives that are a little bit in between. And again, taking advantage, being embedded in a medical environment, we have access to human lungs that are removed by surgery. And so we are taking, for instance, there is a lung cancer surgery, the part of the lung that is not affected by the tumor. You can cut some slices and culture these organ slices for a couple of days, and they maintain biological function so you can do infection studies. And using this approach, you can see, for instance, in the upper panel, there is a reduce in the CT values, means you have more virus as an indication there is virus replication. The first two examples show what you may expect on virus replication. But on the other hand, there are cases like the donor 3, there was no virus replication. So this lung was resistant, even they were exposed to massive amount of SARS-CoV-2 virus. So we were interested, why are patients resistant to infection? And actually what we found, there's a different induction of the interferon response. So you then may look into the lungs, what is the different response, and try to understand what situation makes human lungs receptive or resistant to infection. At the same time, of course, you can also test medicines, how they work in a real human context environment. So it's a little bit in between the animal studies and the real human studies. Yeah, with these few examples on the various elements we try to assemble towards a rapid response platform, I really want to thank, of course, the team who did the work. Julia Riga is sitting here in the audience. She is leading our laboratory. She's the technical head. But also our collaboration we have here with the PATCH group. It's now, I think, 3 years we have. So we had got a wonderful training. But in between, we already have now 4 grant-funded projects running. So you see how this is flourishing. And of course, we highly appreciate our academic collaboration part at Boku University and the spin-off Nova Bioma. And then also, of course, nothing is possible without grant funding. And we also participate in several grants together. With this, I thank you so much for your attention.
Thank you, Kurt, for this great presentation. So please enjoy the coffee break. We will meet again at 11. Just 2 short comments. One is very important for our grant reporting, so please, if you forget to sign the attendance list, just do it. And then, yeah, the other one is just do your networking and enjoy. Thank you. Welcome back, everybody. Welcome back. I have the pleasure to start again this session, but this time it's really me. And this will be about Yoviu virus. So this is it was so funny. I started to make this presentation, and then I was thinking how to put together all this stuff, and then, okay, let's make the title like 10 outstanding questions with Yoviu virus. So I put together these things, and I saw, okay, this is too long, so I just reduced it to 6 outstanding questions. So if any of you will have some questions with Yoviu virus, just tell me. But why? Because we started this whole story exactly 10 years ago. I remember the call from our colleague, a bat researcher, and he went into a cave, and then those bats were hanging there with this hemorrhagic things, and they looked nasty, and then he called us that something is going on which was not detected before, so can you check it, please? And 3 years before, I just asked them to take care of this specific species. This is the Myneopterus bat, because there was already a publication in the database by Spanish colleagues that there is a new filovirus in this species. So we were looking at them, and our first samples were collected at this event, and you can see the same picture at the exhibition. So what we are speaking about now, I don't know how familiar you are with filoviruses, but for sure all of you have heard about Ebola or Marburg. So filoviruses are a group of viruses which are causing a lot of headache, let's say, and damage, especially in particular places in Africa. But the challenge with them is not only the already known members of the family, but the not yet known members and the newly identified members, which we can call novel filoviruses. And there is a great publication summarizing the basic problem with these, that we have a completely incomplete understanding of these viruses, so they can be found in very different ecosystems compared to the Ebola, the classical ones, Ebola, Marburg. We know a lot of stuff about those viruses in Africa, but these new members are like something totally different, including the Yoviu virus, which is currently the only member of its genus, which is the Cueva virus genus. And you can see the picture of this hemorrhagic bat here. This is our picture from the cave. So this is not just interesting because it's the sole member of this genus, but also because it's relation to the nasty ones. I used to say that it's a cousin for Ebola and Marburg, both. And if you check the literature, you can find that not just phylogenetically, but also functionally, it shares some characteristics with Marburg or Ebola. It depends on the processes we speak about. So it is really in between of these. I don't know if you ever tried this website. I really love this. I think this year I will show it to my students, so you can just browse the extremely complex network of zoonotic pathogens. You can click on animals, you can click on humans, you can click on many things. So this gives you the ID. You can see the link on the top right. So you can see the ID of the main questions you might have with the new or suspected zoonosis. So you must understand the place of this virus in this complex network. So as I told you, I put originally 10 outstanding questions, so now it's just 6, but the basic characteristics you must understand is not limited to the lab. It's absolutely super complex, just like with the vector-borne diseases I told you. So the origin, what we know about the origin. For sure we know that it was first identified in this Iberian Peninsula when several caves were recorded with the massive die-off of different bat species, but mostly the Myneopterus bat. And when they checked the history of these die-off events, they found out that there were similar cases years before, which might be or might not be related to this. But finally, after years of investigation, because nobody expected to find a filovirus in these samples, after years and years of investigation, they characterized this Yoviu virus, which was published in PLOS Pathogens. So the main question with the origin is it emerging or endemic? So was this Spanish situation, was the first baby step of this virus on this continent, or was it already here? We have an ID, we have a clear ID now that it shows all the characteristics of an endemic virus. And just 2 examples. We can really find it everywhere where the Myneopterus bats are. So anywhere we go, Balkans, Spain, wherever, it's there. we see the same pattern of the ecological patterns, prevalence, seasonality everywhere. And then if we check the phylogeny of the virus itself, we already see multiple lineages of the virus, which is in accordance with the phylogeography of the host itself. So it is really overlapping. It is an absolute general characteristic of an endemic virus. What about zoonotic potential? So zoonotic potential is a very interesting question because when we isolated, which was a breakthrough, we found that it is easily infecting a wide range of human cell lines in vitro. Okay, that's a nice indication, but still an in vitro is not, it's a good first step, but it really doesn't tell you the full ID. And when we finish the metatranscriptomics, we can see that on human cell lines in vitro, it shows this classical toning down of antiviral response characteristics. And then what else we see is it has very diverse response range between different bat species in vitro, which is very interesting. I mean, not surprising if you work with bat viruses, but still interesting because usually bat viruses are absolutely host restricted. Some of them can be like less host restricted, and just a few are very generative or general in picking the host species. So it really seems to be having a zoonotic potential. But what about pathogenicity? So a key component of filovirus pathogenicity is related to macrophage response. And when, thanks to our Boston colleagues, they tested human macrophage response in relation to UV infection, they found that there is no induction of inflammatory response in human macrophages, which gives you the first idea that this might be the first non-pathogenic but zoonotic filovirus. And then we moved on, of course. So to fully understand the pathogenicity, there is a big step to take, like finding the right animal model. So far, it was not possible. So years and years of research, we still didn't find the right animal model. Interferon knockout mice, ferret, even non-human primates were used, and there is just nothing. So it seems that this virus might be non-pathogenic. If you are familiar, however, if you are familiar with the filovirus publications recently, Rocky Mountain Lab published a very nice paper. Have you heard about Thai forest Ebola virus? It's a mysterious filovirus which was found in a single human case decades ago, and it was isolated from that human case. And during these decades, it was passaged in different laboratories. Same virus, but passaged in different laboratories. So Rocky Mountain colleagues tested in non-human primate model these two separate, let's call lineages, and they showed completely different pathogenicity. So this is now a must for us to test additional isolates, not just the first isolate. So this is what we do now, and we also share the isolate with different labs in the world. Whoever has a capacity which we don't have, like Rocky Mountain has the non-human primate capacity, Robert Koch Institute has a very nice bat laboratory in the Biosafety 4. CSIRO has the swine capacity. So now these experiments are ongoing, and we really try to find a good model and try to understand the pathogenicity of the virus. What about the host range? This is a very interesting question. So thanks to the One Bat Consortium, now we have access to a number of bat cell lines. These are mostly uncharacterized cell lines, so we suspect most of them are fibroblasts, but still they are representing multiple species, which is nice. And we see that, which is again not a surprise, that this virus is really restricted to a lineage, a phylogenetic lineage of bats. So we see that the Vespertilionid bats, the Miniopterus bats are susceptible to the virus. And then the other lineage like Rhinolophus, classical cell lines like Rosettus cell lines are absolutely not. This is again not a surprise, but very interesting. And then this is a generative AI modeling where we wanted to test the GP sequences we have and the affinity to NPC1 receptors in different hosts. And what is surprising, I put too many arrows, but what is very surprising is the shrew cell line here, or the shrew analysis. So that's why we start this year the shrew project with this virus, because there is also an ecological connection, because shrews are occasionally going into the carcasses. So there might be some link or possible link in the nature. We don't know yet, but this is a good way to go. Ecology and transmission, so years and years of research and thousands of samples we took. Now we clearly see that the only sample which can result in infectious dose that you can use for isolation is only the blood. Also, we found the virus in ectoparasites of bats. So the most stable hypothesis is it should be a bloody story. So maybe these ectoparasites can somehow transmit the virus between bats. We are also testing this. We also see that hibernation might be a key in this story because we only find these dead bats after the hibernation. We found similar traits and characteristics to like Marburg virus. We found juveniles with maternal antibodies. So the story is like overlapping a bit with other known filoviruses, but on the other hand, it's a completely different story. So it's a very, very interesting virus. So what is the conclusion overall? We might have 1,000 questions, but one answer for sure, this is a perfect prototype for novel filoviruses. And also what makes it perfect, because this is the only isolated and the only known temperate climate filovirus so far. So this can be a good model also for a very different ecological background filovirus, because in temperate climate, bats do hibernation, which they don't do in subtropical, not necessarily, or tropical regions. So this is very interesting to understand emerging filoviruses, to develop pan-filovirus treatments because of the relation, don't forget the relation to Marburg and Ebola, and also to better understand generic antifiloviral targets. So I hope you might have some ideas with this virus. We are open for collaborations. So this was the 6 outstanding questions. Thank you very much. I am honored to invite Dr. Johnny Ullman as the next speaker.
All right. Gabor, thank you so much. It's an honor to be here, to be invited, and the lab, everyone. I hope I met some of you, but I hope to meet more. It's been wonderful. This is my second time here, and I love it. And I got to meet so many great people. This is great. So the idea, and I apologize in advance, I knew what I should present, but I don't ever do that. I ended up trying to cover as many things as possible because I know that some of the groups that were here, I tried to touch on a lot of things. But mostly, so I come from the United States, from Wisconsin, University of Wisconsin. I'm a disease ecologist, so I do a lot of modeling and forecasts of ecological pathogens. So I called this one Pixels the Pathogens. I use lots of drones, satellite, remote imaging, kind of a lot of big data and high throughput computing. So it's a lot of the kind of the desk-based stuff. But when I go out, I do go out in the field, and I had a blast with Gabor and others in Bangladesh. I'm going to Costa Rica in 2 weeks, and then in Colombia in a month. So I'm going to go through a lot really quickly. I hope that's okay. So overall, what I try to do is look at how can we assess ecological systems, can we understand them with models, and then can we then forecast. So forecasting is sort of the key. Can we, given different ecological conditions, environmental conditions, landscape change, can we predict and understand what might be vulnerable given certain circumstances? So I have training in epidemiology and entomology, and I use ecology and GIS and biostatistics as like my foundation. So I mentioned I use lots of high-resolution thermal drone satellite imagery. There's this new feature thing that we're doing. It's called high-resolution photo stacking. So we use that for macro photography to actually digitize collections and then use AI and other tools to help create sort of this landmark kind of system that people can then use online to have other identifications sort of automated. And then you use a lot of citizen science. So brief overview. I'm only, for the next 2 or 3 slides, it's like 1 slide per project, but I'm working with a professor, Christian Kofley from the University of Notre Dame, and Erica Chomo from KENRI, Medical Research Institute in Kenya. We have a site, there's 20 locations highlighted by these black areas. They're all about 2 to 3 kilometers apart, and they differ in elevation. And what's happening there is that there's kind of localized outbreaks of malaria, and it's kind of constant, but there's heterogeneity. And my side now is to come in and look at the environmental pieces. So we do 2-kilometer buffers, because that's kind of the average flight distance for a lot of the Anopheles vectors. And you can see, so we just submitted an R01, but we're going to be doing some work here in the next couple of years. And then again, I had mentioned in Costa Rica, I'm collaborating with OSA Conservation, which is one of the most biodiverse places in Costa Rica in the southern part, located over here. This place contains 5% of the world's biodiversity, and it is one of the last kind of bastions of conservation in the area. So I'm working with a team, and we're basically going to be assessing the fortitude of the area for impending migrations north of different types of mosquito and tick populations that we're anticipating, which links to another project that I'm doing. So they're highly focused on ecology and protection of their animals, conservation of their mammals particularly. So it's an interesting combination working with veterinarians and biologists and then trying to apply the One Health side to that one. More locally, back in Wisconsin, and it's very cold where I'm from, but we still have persistent and constant tick-borne diseases. We're one of the epicenters in the United States. I'm working with a number of places and colleagues, one of which is called the Marshfield Clinic. It's one of our rural medicine surveillance systems. And I'm not sure if you guys are aware, I'm sure maybe you are, Wisconsin and Northeast United States. So there's a cluster here. This is where Wisconsin is, and then here's the northeastern part. We have some of the worst multiple tick-borne diseases in the world, certainly in the United States. And our Lyme disease prevalence is very, very high. So there's kind of two unique clusters which we have always been trying to investigate. And the part that I combine is a lot of the assessments of looking at climate changes. And we're seeing massive migrations north from different species. So we have the Lone Star tick, Amblyomma americanum, which can transmit lots of other things, especially including alpha-gal syndrome, which is an allergy for red meats, which in the United States, that's a big deal. And then we have Ixodes scapularis, which is our deer tick, the primary vector of Lyme disease, which is actually moving south. Including as well as our wood tick. So our southern states are becoming kind of affected by some of our northern ticks, and then us, a northern state, are being affected by our southern states' ticks. So we have a unique kind of distribution of things here. And then this is to just show that the Lone Star tick range has been expanding massively with climate. In the last 10 to 20 years, it's gone from about here to here, and now it's about here, and that's what we'll be studying in the northern range. And then I mentioned citizen science. We have this initiative is called the Tick Inventory via Citizen Science platform. These were all, we send tick kits to 6,000 people in two years now. And I think it was similar with what, who else was doing that, I believe. We're sending out these citizen science samples, and it's been fantastic. Or Gabor, were you guys doing that? Citizen science stuff with tick kits? I can't remember who was doing that. Sorry. I'll find out who later. But we've been sending these out for the last 2 years, and it's been a great success. We've been able to model all this data. This is all from the tick inventory, from samples from other people. And this has been used in conjunction with our state's surveillance. So we have all this additional data that gets kind of the word out for education, but also for participation. You can see a total of, it's over 12,000, I'm sorry. 6,000 kits, 12,000 samples back. So it's been fantastic. Okay, total random. I got approached by a bioarchaeologist to see if I can do paleoclimatic modeling for ancient DNA, specifically ancient malaria, and this was a project in Athens. And at first I thought this was totally far-fetched, but then as I started diving into it more and more, these paleoarchaeologists are fantastic, and they do this work in the Forum and the Agora in Rome and Greece, and they actually have evidence of DNA from 2,500-year-old samples that actually shows Plasmodium vivax and falciparum. So they gave me that data, and I skipped a whole lot of things, but essentially what we did is we used paleoclimatic data. It's incredible. There was tens of thousands of samples all across the world, and we used those radiocarbon isotopes from soil, from trees, from insects, from all these different things, And we basically could kind of sort of have a relatively accurate assessment by month for 20,000 years in the past of what the atmospheric conditions might have been. So we applied basic epidemiologic models in accordance to what the primary vectors of malaria might have been, and kind of still are actually. And this is just a cumulative assessment showing from 1000 BCE to 1080 how many months are suitable for malaria transmission. And this is going to go for a long time. But you can start to see this relationship where, you know, the Mediterranean areas, but in particular the Balkans, gets kind of warmer than expected, which is really interesting. I think a lot of that has to do with the ocean's coasts in the jet stream. But here's another, I think, a better image to show kind of the modeling element of this, where every month, so every dot you see is each year, and each year shows what those climatic conditions were. And that yellow line at the top shows the northern limit of suitability. But the red line at the bottom here, this shows kind of the endemic line where we see constant malaria is what we'd expect. And sure enough, so like Athens is right here. It's right at that cusp of at least 4 to 5 months of what we expect transmission to be. But also, coincidentally, is pretty interesting is all the other area around it up until this line is in theory suitable for at least 1 or 2 months. So not that long ago, malaria was much more prevalent, and it's, in theory, could be. So I didn't show the slides, but a part of this is to understand what we might expect going forward. And in the U.S. in particular, we're experiencing local autochthonous malaria transmission increasingly more common. I'm going to just fly through. I'm going to move on faster because I want to talk about Bangladesh. So I did talk about some of the work I'm doing a little bit in Costa Rica. This stretches down to Colombia. Has anyone heard of Oropouche virus? So it's kind of a re-emerging disease. It's an orthobunia virus. It's actually spread by midges, biting midges, which are literally these incredibly small but complicated and very understood vectors. It has an urban and a sylvatic cycle, and it's really kind of exploded in the past decade. Here's the range of the primary vector, Culicoides periensis. It's spread all throughout the Americas. Given the size is a culex, I think quinque fasciatus. You can see the relative size. They're literally called noceums because they are so small and they just kind of dive bomb, get blood, and leave. In the last 2 years, in the larger South American, Latin American countries, we've seen a massive explosion, this kind of bimodal curve of cases. And it's really become more of an issue now and more widespread. And just recently, there's been 2 deaths in 20-year-old females, which has been, maybe there was deaths for a long time, but this was the first time it was recorded, so it's starting to catch fire. So I'll be doing some more fieldwork assessments. In this part, this is Leticia, Colombia. If you can kind of tell from the inset, it's located at the very tip of Colombia, right on the Amazon River. It's a very pristine jungle kind of ecosystem. And the reason we're going there is the diversity of pathogens from some early sampling. We have a lot of other ongoing things happening. So in addition to Oropouche, we have all kinds of malaria right now. Yellow fever is recent. And then, of course, dengue's been out of control really bad. So it's sort of an expedition of finding what can bite us. And the cases where, even though it's a small place in the middle of the jungle, there's been a high amount of cases at around this time last year. So this is just looking, it has a north, kind of south urban gradient. And some of the satellite imagery I've been looking, we look at disturbance. So this shows where human involvement has been, as well as what does the ecosystem largely look like. So we can see where built environment is, where jungle is sort of recovering or being destroyed. And those are strong indicators of where to kind of look. This is a conglomerate of a lot of different things, but this just shows some of the value, some of the satellite data we can get. It shows not only water or land, but it also shows how wet something is, which can be very important for Culicoides habitats. And then these are the field sites. So we're looking at the urban, very dense kind of urban area, and then all the way out to a very jungly rural place where there has been previous cases. So this is kind of along the water riverine edge. All right, finally, I have 3 minutes to talk about Bangladesh. So this is a really exciting, kind of the really fun thing that brought me here ultimately to Gabor. I call it modeling Pteropus medius ecology, but it's really to help understand the Nipah virus ecology alongside the rapid testing that we're doing. So this is the Swiss cheese model adapted from Raina Plowright, a colleague of ours. So what we're hoping to do is we have this haystack, we're trying to find a needle. So our haystack is a bat roost or multiple bat roosts, and which one or two or whatever, how many of those bats might be actively shedding at that time. And for that to happen, we have to have all these factors fall in place. And it's pretty phenomenal that transmission happens. And then we find out that how many people are being exposed, that this transmission doesn't happen all the time. It's incredible, which is a fortunate thing. So again, talking about all the different pieces I look at, I do lots of environmental satellite data, but I also do socioeconomic and ecological pieces that all kind of fit into this larger piece. I mentioned the drone. I mentioned high throughput machine learning, which is like a supercomputing cluster, which helps us process all this massive data. But I'm gonna talk about this fun toy. It's a tool, but I call it's a toy for me. It has this incredible thermal image camera that just came out, and it's one of the best on the market. And it exceeded the expectations. This is the video from one of our roosts that we went to. This is actually one of the larger ones. All those little dots that are glowing is a single Pteropus bat. And in this one, there's not just one tree, it's like the whole forest there is sort of, roost. This is one roost of thousands of bats. And we can do lots of things with this data. The data is embedded, so it's very high res. We can get really close, about 10 meters, and then once you start getting there, then the bats start talking and getting kind of nervous. But it's incredible, and then also it has a 15-kilometer range, so we can actually follow the bats. The only problem is my battery doesn't last as long as the bats fly. So we have some other options that we're exploring with some low-cost GPS units that we should be able to hopefully get and put on at least 10 bats per roost is the goal. So the drone is very powerful, and it also has embedded thermal technology. So we can see individually which bat, what temperature they are. And in theory, I'm having AI, also my students, unfortunately, doing the guinea pigs. We're counting each bat, and we're also assessing which one might be statistically high or low, which might be indicative of illness or febrile, et cetera. So this part is really slow, it's terrible, but manual annotating is not fun. So we're trying to do these well-trained AI modeling. And this is just this is pretty new right now, but we're doing a comparison of side-by-side of thermal and real-time real imaging. And great news is that the thermal is very strong. Our model is picking up the video real-time counting, which is excellent. The image that's not thermal, which is just like looking at it, is very difficult to assess. So the thermal piece is an essential critical tool that I think we're going to be able to do a lot more with the ecology side, especially as we get more and more roosts. And then incidentally, we found, accidentally, one of these capped langurs, which was a very rare, I guess, primate or monkey species. So that was excellent, and that was exciting for me because I'll be taking this same model approach to Latin America, which was the key there. And then this is the recent assessment that we got from an overview of the 3 years of collections that Gabor and his team had been doing. This shows the distribution of where those roosts have been. That's sort of the NEPA belt, the hot spot of where NEPA is. And then alongside it is the human deaths per 100,000. So you can kind of see the change in the distribution of where things have been and where stuff is mostly affected. And we've revisited quite a few sites, I think, but some new ones in there, and then we have some other new ones, which is exciting. And then I'll just kind of hopefully go through this quickly, but we're hoping to integrate some citizen science to help us understand what date palm trees, the primary tree that is where we think the culprit of transmission is happening. The drone can tag them, but the drone can't be everywhere. So what we're hoping to do is get people to help us passively just tell us where, we saw a date palm tree, we saw somebody harvesting. And this is in development. We created this QR code that has this bat observation survey that we can submit basically an image, a location. Anyone can do it with their smartphone. And surprisingly, everyone is really connected there with their phones. And then this is another example. I built this global mosquito dashboard from Citizen Science. There's something like 500,000 observations. This collates all known mosquitoes and hopefully expand it to ticks one day. This is a massive project, but this just shows high resolution images from anyone, and this is all free data that we can use and leverage. So those high-resolution images that I referred to, I'm trying to also integrate modeling AI approaches to help us identify what those mosquitoes are in real time. So we have these little mass-based morphologic regions. And here's some more examples of some of the modeling. And from Aedes aegypti and Aedes albopictus, we have those kind of beta models up. It's not perfect, but it does a pretty good job. So this is the photo stacking. We actually take our insects of interest, put them on a camera, and then we have a 360-degree camera, high resolution, that goes all the way around, automated, and it puts it into one big model that we can then use as a sort of the Rosetta Stone for other images. And then I'm working with Arubata in the UK, which is helping us with these low-cost GPS units, which I'm excited, very excited to hopefully attach to some of our bats in the future. That'll be really helpful. And what's interesting about this company is that they're nonprofit. They try to do low cost, so 150 US dollars or less is what they estimate this 15-gram unit to cost. So very exciting, I think. And it's not a telemetry tag, it's an actual GPS unit that gives us like real-time location data. Okay, that was a lot. Thank you. And please, we can talk later if you'd like. And I'm here to collaborate and open with lots of different things. So thank you so much. I went long.
Thank you, Johnny. It was great. I don't know what's your experience with virology conferences nowadays, but I think that one of the hottest topics are related with viral persistence. So please, Professor Miles Carroll is the next.
Thank you. So a real pleasure to be here. First time I actually was here was in May 2018. We came over to Pete Mays in the red jacket, and obviously Frank and Gabor. We came over to try and help out on the Yovovirus when people wouldn't initially believe that you had Yovovirus in Hungary, which was a great piece of work. And obviously temperature's a bit different now, but still lovely to be here. So today I'm going to talk about some work we've been doing for the last 10 years, really, assessing long-term immunity to Ebola virus and survivors in West Africa. And then something more interesting to me in recent years is how Ebola virus and other viruses are able to persist in humans after recovery from symptoms. Quick bit of background in my lab at the University of Oxford within the Pandemic Sciences Institute. So three main themes. The one on your left covers a lot of what Gianni and Gabor and many others do on looking at surveillance of pathogens, primarily in West Africa, but also in Borneo, and also in bat populations in the UK. On the right, we do a lot of antiviral work, so it's lovely to see all the chemistry. Honest, I love the chemistry. And work from all the region and had some good conversations with colleagues on that. But what I'm going to talk about today is the middle section, and that host pathogen interaction and immunity. So we've already heard about filoviruses from Gabor. So negative sense RNA viruses, the first outbreak that we know of in Africa was in 1976. Obviously, in 1967, there was a Marburg outbreak in Marburg, but we think that's from non-human primates that were imported from Uganda. So obviously, since that time, there were numerous outbreaks of many hundreds of people, until a major epidemic actually occurred between 2013 and 2016 that started in Guinea in West Africa and rapidly spread to Liberia and Sierra Leone. We were important down when I was head of research for the UK Health Security Agency. We were part of the European Mobile Laboratory, which some of you may also have been members. And we sent 50 of our staff with many other Europeans to work in the diagnostic centres and the treatment centres throughout those three countries of West Africa, but we primarily funneled a lot of our energy into Republic of Guinea. But it just shows to show that in a relatively short time, when you do actually get samples out of an area that has an unexplained sort of death toll from hemorrhagic fever, that you can instigate a mobile laboratory pretty rapidly. And obviously, this was back in 2014. So initially we were providing Altona quantitative PCR pan-filovirus, then we zoomed in on Zaire, as that's what was found to be the culprit. This was all very good for MSF and other colleagues that we interacted with in the treatment centres in who to bring into the treatment centre and when to let them out when they recovered. But we quickly retooled and we provided real-time sequencing data using the MinIRN when it was in its early days, and this is obviously some 10 years ago. It performed phenomenally well. We had turnaround times that were better than the public health facilities in the UK, and we were doing this out in West Africa. So we provided this real-time sequencing, which obviously covers people called molecular epidemiology, and we found some really interesting chains of events that occurred that the classical epi or Shulev epi wouldn't have identified. So I don't know if there's a... we had up in a region of Gekadu, in the forested region, about 600 kilometers up from the capital is where we were working. And this was 6 months after the last case. So we were bleeding survivors looking at long-term immunity. And we found a death in there was a young lady found dead in an area near us. Luckily, a smart healthcare worker managed to take a sample, showed it appeared to be positive for Ebola. The sample then was sent down to the European Mobile Lab, and we confirmed it by PCR as positive, and then by sequencing showed it was a really strange result, that it was very similar to a gentleman that we'd released from a treatment centre that we'd managed to sequence 500 days previously, which is very, very strange, because there should have been a number of mutations that should make this person a little bit different at least. Anyway, the gentleman was tracked down, asked if he knew this lady. He obviously said no, but then he gave a semen sample and a blood sample. The blood was negative, semen was positive, the sequence was a direct match. And so with that information, you go back to the individual and ask them, did he know this young lady? And then he admits that he knew her and was having a sexual relationship with her. So the virus is obviously persisting in this survivor, most likely obviously in his testes and shed in semen. But it just goes to show that molecular epi, how quickly it could turn around the background of the new flare-up. Because initially he thought it was a reintroduction by bats, but quickly we found that it wasn't, and it highlights the danger of male survivors instigating these flare-ups. So WHO, with that evidence, set up a vaccination schedule around all male survivors. So those immunologically naive women would have a chance of protection against sexually transmitted Ebola virus. And so that worked pretty well for 5 years. Another case in 2021, back in the same region, we found that another lady that had died in the community, she managed to transmit to a number of her family members, but they sequenced the family members and showed that it was phylogenetically very, very similar to a male survivor from the 2016 flare-up. So 5 years is suggesting that the virus has been persisting in the testes of the male survivor. So we recently published a review on virus persistence, and a lot is actually known about it. People have been describing issues. We knew that Ebola virus could survive in male survivors' testes for at least 3 months before the epidemic. But obviously with much more data, much more men that have become infected, the more analysis we could do. And there's been cases of infections in the eye and recurrence of disease from there, in the brain, another recurrence of disease in a returning nurse to the UK. This was a US doctor. Numerous cases of virus in mammary we found in West Africa and also in vaginal secretions, but the testes has the highest sort of hit rate. And connected a bit to an earlier talk on hantaviruses. So we know that it's a case for 6 years that a person that recovered from hantavirus infection while he was in Chile returned to Switzerland and they found a virus in semen 6 years later. So if you model all the incidents of the publications on virus in survivors' semen from West Africa, you pull maybe 3 or 4 of those models, data sets together and remodel it, it comes out that there's about 6 or 7 male survivors that would still be shedding virus 5 years later. So we know that's partly right because we found one in a case back in 2016 individual. But there's obviously many more out there, so it's not 100% efficient to infect a naive female and cause a sexual transmission flare-up. So we know from publications on non-human primate studies that the Sertoli cells that are the margin between the testes and the blood barrier here have been shown to have long-term infection with Ebola virus. So that's obviously somewhere where we're zooming in and trying to understand. But just go back to a couple of key points here. We know that the virus has a much slower mutation rate when it's in the testes, and this might suggest, logically, that the virus is replicating in slow motion. But how is it doing that? There's no molecular signatures in the genome that would give you any insight into that. So I used to have a CL4 facility when I worked at Porton Down. We don't have one anymore in Oxford. We have good CL3. So looking at CL2 alternatives to working with Ebola virus, and we linked up with Pete Mays, who I showed in the earlier photograph, who's now at the University of Brussels. So he uses the VP30 Delta Ebola virus system that was first developed by a group in the States some 10 years ago. So VP30 is an essential gene. You knock it out, you provide it in trans in your helper cell line, and you've got this biologically contained Ebola virus. So it's taken some years to reduce that down from CL3, and now we can use it in Europe at CL2 in many European countries. So working with some extremely good cell biologists in Oxford, Fabri Rath, he derives iPSC, so skin cells pushed down to different lineages, and we can remake a human testis in a Petri dish with the 7 key cell types, including Leydig cells and Sertoli cells. And the idea is that well, we can do this now, but we haven't got VP30 in there yet. That's what we're in the process of doing. And when we do that, we will then try to repeat the environment in the testes, which obviously with different pH, probably different temperature, different oxygen levels, and see how the virus can, if we can slow it down, and we'll look at other signatures of persistence. So we already know that Sertoli and Leydig cells infected with Ebola virus, this was done at the defense labs at Porton Down, completely obliterates the cells rapidly. So that can't be the mechanism in normal normoxic oxygen levels, etc. So we need to manipulate the environment to make it closer to reality. So then a little bit, so very much linked to this virus persistence is what role it plays in inducing long-term immunity. So there's lots of viruses out there and lots of vaccines like yellow fever or smallpox vaccine that are really give long-term immunity. And there's no evidence of virus persistence for those vaccines. But what about Ebola? Could that virus be being shared? Could it re-stimulate the immune response? So we've been working with the survivors of Ebola virus disease in the Gekadu region. And what we've looked at is over 10 years now, this is the first 4 years, of their antibody response that has a steady decay rate, but still pretty strong. If you look at the neutralizing antibody response, Florian showed for hantavirus, we do see an increase, but then come down. And that's known in maturation of antibody responses. When we look at T-cell responses, looking for signatures of re-stimulation, so memory phenotype is always distant memory, so not that they've been re-stimulated at the T-cell level anyway. This paper came out in Nature in 21, suggesting from analysis of a cohort of Ebola survivors from Sierra Leone, looking at them from day 120 to 500 post-recovery, that they said that in a few of their individuals, they got this peaks and trough of antibody responses. So, and they said this could be explained by persistent virus being shed into the blood, though we didn't really believe that. However, could be right, so we worked with a fantastic mathematical modeler, Kim Fornes at National University of Singapore, and just applied the classical decay formula. So their decay rate was 6 months to 2 years, a t-half. Ours was 10 years, somewhat different. And we looked for peaks and troughs of our antibody response over the period of time, and we never saw it. So we now have 10-year data. We went back last year, and it agrees perfectly with Kim's original model from the data from the first 4 years. So the longevity of immunity to Ebola is exceptionally good. So then in summary, I mean, for us, the Ebola virus persistence creates this additional mechanism of outbreaks. I don't know if you can call it a spillover from another human, more than a human-human transmission. And understanding those mechanisms is a really important public health question for preventing not just Ebola virus, but we know a number of other viruses can also persist in the mouth testes. And with that, just to highlight the key collaborations with Pete Mays, my Guinean colleagues, Raymond Akoy-Bore, and the key immunology work was done by Tom Tipton, and virus persistent work with Ola Diebold. Thank you very much.
Thank you, Mais, for this great presentation. So the next one is Maurice Bardsley. He will present us the EVA project. Thank you.
Morning, everyone. Still morning. I'd like to thank Gabor and Balázs for inviting me here. I'm not quite sure what I'm doing here because I know nothing about viruses, but what I'm going to do is talk to you about the European Virus Archive, which is a resource for researchers. Just to give you a bit of background, even though I'm affiliated to Aix-Marseille University, you can probably tell I'm not French. I'm the only non-French person in the management team in Marseille, but I'm based in the UK when I work from home. So I'm actually retired from full-time work, but I work as a consultant to the EVA project in terms of quality management. I've had two careers in my background. I started off as a materials chemist, designing acoustic stealth coatings for nuclear submarines for about 25 years. And then I fancied a change in that sort of role, and I took a job at the Veterinary Laboratories Agency in the UK in Weybridge, where I was managing a biobank of prion materials, which was to supply them to researchers and test development companies across the world for diagnosing a human form of mad cow disease, Creutzfeldt-Jakob disease. During my time at the VLA, I became involved in the creation of the EVA project in 2008, 2009. So what I'm going to do now is just talk you through what it is. So the EVA Global Biobank, why was it needed? Now, even as recently as the first 10 years of the 21st century, it was difficult, and I'm sure you'd agree, that it's difficult to access virus collections unless you're actually involved collaborating within a virus club where people had viruses that you could access for your own research. So EVA was created to try and make a change in this to support research and diagnostic development and vaccine development. Now, of course, there were collections of viruses existing, but in those days, they weren't well publicized, there was no coordination, they were variable quality, poor biobank management, which thankfully has improved dramatically now. And there was a degree of protectionism. People didn't really want to share their viruses because they didn't have any strong perception of the benefits of sharing. So this is the original biobank consortium that started off under Framework 7 funding for EVA. So these are 9 European labs who started off And it was originally created in Aix-Marseille University with the likes of Xavier de Lamballier, Remy Chirel, Ernie Gould, sadly passed away now, but he was an ex-professor of virology at Oxford, and Bruno Coutard. The vet labs with Tony Fuchs and myself, now APHA, which is when we were in Europe in the past. Miles Carroll's lab, UK Health Protection Agency, now the Health Security Agency. University of Ljubljana with Tatjana Avcic and Boris Klemper in Slovakia, and Daniel Pinchware at the University of Geneva, now at Basel, and the Bernhard Nocht Institute in Hamburg with Stefan Gunther with his 2 BSL-4 labs, and the people you well known, of course, would be Christian Drosten, Drexler, and Marcel Muller, who were in Bonn at the time, now in Le Charité in Berlin. So all of these people started off willing to share their viruses with researchers in the virus community. So thankfully, because I have a background in quality management, even though I have a scientific background, I have a lot of experience in quality management systems. The coordinator at the time, Professor Jean-Louis Ramette at University of Marseille, was very supportive of a quality management approach to the EVA system. All the partner labs were required to comply with the standard, and what happened at the time was I looked around, there were no ISO standards which actually hit the nail on the head in terms of what we were looking to do, which is to open up access to high-grade, high-quality virus reference samples. So I wrote a standard based loosely around ISO 9001, which is about process control, but with a difference, which was in fact the way in which we were grading the viruses in terms of identity, sequencing, partially or full sequencing, purity, looking for mycoplasma contamination and purifying where necessary, viability, looking at expressing the infectivity according to the way in which each lab would express it, and stability as well, preservation in storage. So all of these labs committed to following this quality standard that we implemented within the EVA project. And I would then go along and audit them, but I did it in a friendly way. So we'd always stress that we're not looking to catch people out, but we're looking to see if we can share best practice because we've been looking at other labs and we're sharing what other labs are doing, and we're looking for areas in which they can improve their quality management systems in their own institutes. So then, in the next funding period, we became a kind of victim of our own success. So EVA became EVA Global, where we opened up, everybody wanted to join the party. So we were then increasing in our membership. In terms of the products, it's not just viruses, but also derived materials, proteins, nucleic acids, antibodies, and so on. But they were all quality graded, and this grading is put up on the website, which is publicly available. Platinum is the highest grade at the moment, fully sequenced, purified of mycoplasma and infectivity expressed, and either lyophilized, freeze-dried, or stored under -80 or under liquid nitrogen. So this gives you an idea of the size of what EVA Global was. These are all of the institutes and labs that joined the party after 2015 to about 2020 under the Horizon 2020 funding arrangement. So it's not just human viruses, but also veterinary viruses, but also plant viruses. So we had Julius Kuhn in Braunschweig, DSMZ in Braunschweig had plant viruses, ANSYS in France, and the French Food Safety Authority. And there was also the lab in Turin with Gianpaolo Accotto. But also we had University of Florida and the lab in Singapore, although the Singaporeans never quite joined the party, but they were dancing around the outside as an associate member. But all of the rest of these, the FLI, Le Charité, Robert Koch Institute, were all active partners uploading their data onto the database and actively operating and supplying to research groups. Now you might think, well, why can't we have all of this in one lab? Of course, logistically that's not possible. because there's a staff resource and the physical size of an archive for all of the virus products. And there's still an ownership culture within the project. So the partner labs within the project are free to make their own decision. They curate their own collections, and they decide what they want to share, what they want to upload to the database. I find that coming into this world, there is a very strong team ethic. I think I've met a lot of really nice people, and it worked really well within the project. And there's a paradigm shift I've noticed in the culture, in the culture of virus sharing. So the consortium labs also complied with the project QMS, and I've updated the project standard over the years to improve it and to involve biobanking standard 20387, 17034 for reference material producers, and I'm also going to be updating the standard soon to reflect things like biosafety 35001. So researchers are free to interrogate the online catalogue. It's publicly available. Nothing else than that. If it's there to be seen. In the days of EVA Global, there was the ability to look for transnational access supply. This was free supply. They just have to pay for the courier costs. But the EC compensates the EVA lab for the preparation costs for transnational access. And that looks at about 50% of our dispatches during the time. You just need to be a bona fide publicly funded lab to receive free supply. And of course, we've also supplied industrial pharma companies. Something like about 30% of our dispatches are to pharma companies. You'll be familiar with all of this, of course, but this just to give you an example of what happened in the MERS coronavirus outbreak in 2014. The EVA Global project supplied control materials to 151 labs across 54 countries with collaboration with WHO regional offices. And during the earlier phases of the MERS coronavirus outbreak, EVA distributed in less than 2 weeks diagnostic materials, including 300 PCR test kits. Then, of course, there's 2014 Ebola, Zika virus, yellow fever, coronavirus, and monkeypox. So this gives you an idea of the activity in EVA Global during the coronavirus outbreak. And at the height, within the first 100 days of 2020, we were receiving something like 200 to 250 requests for positive PCR controls every week. And these will be usually for free supply transnational access, or from publicly funded diagnostic centers across the world. So we receive requests from Russia, Iraq, Iran, Colombia, as well as European labs as well. Just slightly mischievously, the picture you see there, the Chinese guys, I took this photograph, so I know it's real. It's not AI. And these guys were allowing me access into their freezer store because the Wuhan Institute of Virology was an associate member of EVA Global, and the Chinese government gave them something like $1 million funding to participate. We didn't get very much, maybe just antibodies from the Chinese, but there was a much more interaction than if we hadn't involved ourselves with them. This gives us a screen cap of the database, just to give you an idea of what it looks like. I'm sure many of you have looked at it already, but you can see the viruses listed on the left-hand side, and you can drill down on each virus product to look at the data associated with it. So there's a complete provenance of the data, the cell culture used, the storage capacity, the storage conditions, and also the grade, whether it's a platinum or gold standard, and the price. In the past, some labs prefer to pay rather than having free supply, and that's because they don't want to share their scientific work with the project. So in order to receive free supply, you need to supply a scientific justification for receiving the virus. And once I've cleared as the quality manager for the project, I've looked at the documentation that approves the facility and the expertise to receive and handle the virus, then that then goes on to an external scientific selection panel, and they look at the science underlying the request. Most of the time, 95% of the time, they've ticked it off and said, yeah, that's fine. Mostly it's for developing a diagnostic, but there's also research as well going on. So on the catalogue, we are at the moment, because I'll get onto it in a moment, but we are going through a bit of a transition because we've just become a legal entity as EVA AISBL, so we're sort of recalibrating ourselves. But on the catalogue, it's not just about virus products and derived materials, but in the past, we've also provided access to training for PCR diagnostics, sequencing, infectious subgenomic amplicon, reverse genetics, and BSL-3 access. And we've had a lot of African countries, African labs coming to places like Pasteur in Paris, the APHA in the UK, and Marseille, and German labs as well. Some numbers for you. This is for about the first two funding periods over 8 years. We distributed over 7,000 products worldwide, about 50%, as I said earlier, as transnational access. So the rest of it is fairly explanatory. 30% to industry, and many references. And we've got something like 2,900, almost 3,000 virus strains on the catalogue. Not all of it is currently visible because we're undergoing this transition, so there will be data on the database that's not visible because the owning lab are not rejoining EVA AISBL at the moment, but that's changing. So what happened last year? We've become this legal entity, and I need to pay tribute to Professor Jean-Louis Romet, who's now retired and lives in his alpine lair in the French Alps. He's now the mayor of his village. But Jean-Louis was very motivational, and he had great vision and leadership. And we always, together, we would always interject a degree of humour and teamwork that's developed, and he's responsible largely for the success of the EVA project. Bruno Coutard is now taken over as the coordinator for EVA, and EVA, as you can see, is also part of the Evora project, where we're working with Kurt within the quality management work package within Evora. So Evora is Arena, EVA, and Elixir for the data management guys. So this gives you a picture currently of the membership of the EVA AISBL. AISBL, if you're not familiar, is a French acronym basically meaning not-for-profit. So these are all the original European labs, plus some of the EVA Global, and the only one that's not European is Casero in Geelong, outside of Melbourne in Australia. And literally 2 or 3 days ago, Erasmus Medical Centre and INMI in Italy have just rejoined. Basically, the requirements for joining this EVA AISBL is that you need to provide a political statement from your national authority or government department to say that you're prepared to comply with the terms and conditions of joining the consortium, and also a financial statement saying that you're prepared to pay the subscription. At the moment, it's about 7,000 euros for a full member, which gives you voting rights to admit other members, and about 50% of that, 3,500 euros for an associate, which gives you equal access to the database. And this, just a little shout out for my days in prion science, just to close off. So thanks for your attention.
Thank you, Maurice. So we have 2 more presenters, but unfortunately, due to administrative issues, they could not make it in person. So we will get them online. And the first one will be Professor Nitish Debna, and I'm particularly proud to know him because he is our biggest supporter in our activities in Bangladesh. He's a professor of microbiology and public health, and He is taking part in a number of international organizations as a chief scientific advisor, and Bangladesh is a special place because it's among the few countries who has One Health as a term and an activity on the administrative level of the governance. And the key person to achieve this was Professor Nitish, so I'm super happy to have him. Hello, Professor. So the floor is yours.
Thank you, Gabor. Can you hear me? Hello?
Yes,
Can
yes,
you hear?
please, you can start.
Yes.
Thank you.
Okay. Thank you very much for your kind and nice introduction. Ladies and gentlemen, basically I will not talk about viruses or other things. I will be focusing on how we have developed our One Health approach in Bangladesh. And that will be my subject of discussion or talk. And here is the um, uh, slides are not moving, are they? Right. Something wrong? Yeah, I think, can you see my
Yes.
slides now?
Yeah, the slide is visible now. Yes.
But do they move or not? I am trying to move them, but it is not moving. That is the issue that I am struggling with.
Még kétszer megpróbáljuk, és akkor utána mondom neki, hogy... Professor, if you can't share the slides, we can share it and we can
Please share from your end, and then I can go ahead.
Okay, so we will share it, and then you just need to say next slide, please, and that's it.
Yeah.
Okay, thank you. So it is shared now, so you can start. Thank you very much.
Okay, ladies and gentlemen, basically what I'm going to talk about the One Health journey in Bangladesh. Next slide, please. As you can see in my next slide, here it is all about the outline of my presentation, and that presentation gives you a bit of opportunity to what I'm going to talk. And basically, I will give you my slides. Something wrong here, I don't know. About can your other person talk? I can come later on. You can uh, put me later. Hello? Can you hear me?
Yeah, we can hear you.
Okay, Professor, so we will jump on Rafael's presentation. So in the meantime, you can prepare, and then we will try again. So please be online.
Yes.
Thank you.
Yeah.
So I will give the floor right away to Dr. Rafael Elias Marquez,
Yeah.
Structural Virology. Thank you, Rafael. Please start. Thank you.
Thank you, Gabor. Thank you, everyone. I'm going to just move this to presentation mode in a second. So just let me know if you're seeing the actual presentation or the back slides.
It's not the presenting mode. Yeah, now it's good.
This should be
It's
correct.
good now.
I'm going to have my pointer here, and I understand that maybe some lagging. If the presentation somehow is not moving, you guys just let me know.
Okay.
So, Gabor and friends at the VNL, thank you very much for inviting me. It's such a pleasure to be part of this celebration. And as you mentioned, it's quite sad that I'm not able to be there in person to join all of you. And I'm going to take this 10-15 minutes to tell a story about St. Louis encephalitis virus that we are working on. I'm going to show some unpublished data. And how can we draw insight from the structures of a neglected arbovirus that circulates here in the Americas. So I'm the leading researcher in virology at CNPM. And I'm going to tell you just a little bit what CNPM is. So this is a research institution in Campinas, in Brazil. And one of the reasons why we started to communicate with Gabor and our friends at the VNL is because we are moving forward in the past, I would say, 5 years into researching containment, in biocontainment. So we already had a BSL-2. We built a BSL-3 recently, which is located here in the Brazilian Biosciences National Laboratory. And in the near future, we will conclude Project Orion, which is going to be the first BSL-4 in Latin America, which is to be constructed right here next to the synchrotron source, a particle accelerator that we have here at CNPM. So we plan to connect the these two very complex research infrastructures in order to be able to do cutting-edge imaging experiments inside maximum containment, which we think is something desirable, and we want to spearhead this initiative globally. So going back to the subject of the study that we are doing right now, St. Louis encephalitis virus is a neglected mosquito-borne orthoflavivirus, so related to dengue, to Zika, to yellow fever, and other human pathogens of interest. Different from dengue, St. Louis encephalitis is transmitted by Culex mosquitoes and has its reservoir in birds, which is very important for the ecology of this disease. So if you get infected with St. Louis encephalitis virus, you may develop this severe neurological disease, which is named as the virus, and that usually starts as a febrile illness that evolves to this severe neurological disease. There is a higher risk in the elderly to manifest the severe forms of disease. And being a neglected virus and a neglected disease, there is no vaccine candidate or specific treatment licensed for human use. So we're pretty much in the dark here regarding this disease. There are several papers from different groups. Those two are from Lark Coffey's group in California. We have groups in Brazil showing this. These are the most recent papers. And also in Argentina showing that the virus circulates in the entirety of the American continent, from North America to South America and through Central America. And altogether, we believe that St. Louis encephalitis virus is re-emerging, which is something worrisome. So this is a model by Kopp and collaborators from 2013 showing how this virus can circulate in the Americas. So because birds move north to south and vice versa, those reservoirs carry and spread the virus throughout the continent, and this is quite let's say, dangerous, but also interesting to see how the virus evolves as it moves around the continent. So we have some background work on structural biology of viruses. We were the first to elucidate the structure of myelovirus, which was the first structure of a virus ever elucidated in South America. So we took that background and a suitable target, which is St. Louis encephalitis, and we tried to combine that in order to really provide the information necessary for the development of disease countermeasures against St. Louis encephalitis. And there is also this special point right here, which I want to mention, is that we already know, obviously, how to work in BSL-2 conditions. We made huge advances since COVID-19 on working on BSL-3 laboratories, and we really want to move forward expanding our capacity for structural virology in greater levels, higher levels of biocontainment. So eventually moving to BSL-3, which we're already doing, and as Orion is inaugurated, we should be able to do that in BSL-4 as well. So how we do it? Inside biocontainment, we propagate the viruses in huge titers, in huge volumes. Here I'm using this figure, which was made for myeloid, but for St. Louis encephalitis, luckily the protocol is very similar. And we just propagate those viruses up to, let's say, 2 dozen T150 flasks, meaning that we're going to harvest around 500 to 600 mLs of infected supernatants. We then precipitate the viruses and do 2 rounds of ultracentrifugation, again, all in containment, to yield samples that we do quality evaluation on, assessing the virus titer and assessing virus morphology. And if this is confirmed, we prepare electron microscopy grids, which are read and analyzed in a microscope like this one, the Titan Cryos G3i. The Titan Cryos is not located, at least not ours, is not located inside the BSL-3 like it is in some parts of the world, but we're able to, in some instances, to inactivate more dangerous samples to bring it in. Fortunately for St. Louis encephalitis, this wasn't necessary because the environment around the laboratory is considered BSL-2, and St. Louis encephalitis is a risk 2 agent in Brazil. So we prepared samples according to this protocol, and yielded results like these that I'm showing in these images here. So what we're looking at in these first 2 pictures here is a negative stained preparation of St. Louis encephalitis observed in the transmission electron microscope. So as you can see, St. Louis encephalitis has typical orthoflavivirus features. We have around 50 nanometers in diameter virus particles, and as we were able to confirm that morphology and quality was enough, we worked on concentrating the samples because this is a requirement for cryo-EM analysis. And we eventually reached a sample that looked pretty much like this, a very crowded image of purified viruses. And then this type of sample is prepared in cryo-EM electron microscopy grids. And we're hoping always to have these little black dots, which are all virus particles landing in these spots that we call here, which are then analyzed under the cryo-EM microscope. So in order to have these many viruses, we have to reach titers as high as 10 to the ninth power plague form units per ml. And in this case specifically, we used viral CCL81 culture, so we're producing this virus in mammalian cells. So this is what we found. So we were able to collect many images of St. Louis encephalitis virus particles, and as we combine and process those data, we reached a model with a very high resolution, 3.29 for the entire virus particle. And I'm gonna play here, and you should see this virus particle moving to the right. So it's spinning. And what you're looking at are in gray are the E proteins and the very surface of the virus particles. Inside you see the M protein, which is part of the virus scaffold, and a lipid pocket behind it, which we also want to investigate. Because we achieved such a high resolution, we're also able to see the glycosylations on the E proteins at the virus surface, which is colored here in orange. So we're very happy about it, and as for you in the audience that are experiencing this subject, glycosylations are crucial for almost all viruses that we have ever worked on, especially those causing disease in vertebrates. So we're investigating this, but not only, and this was a very curious part of working on St. Louis encephalitis, is that as we are analyzing the images, we see this class, this particle class here, which matches the shape and size of a mature virus particle. But we also observed something like this on the right. So we have those viruses that have a more, let's say, spiky conformation. And at first, some people working this project thought that we had a contamination, but knowing about orthoflaviviruses, what we're looking at was actually the immature form of St. Louis encephalitis. And this is very typical of orthoflaviviruses as well, but we're not expecting to find as many mature viral particles in our preparations. And because of that, we're able to elucidate both structures at the same time. So this is the structure of the immature virus particle. In green, we have the same E proteins that we're seeing in the first slide, and the glycosylations here are marked in red. As you can notice, the shape of the viral particle is so different, and the proteins are so differently arranged that the glycosylations that were lined side by side in the mature virus particle, in the immature form, they are at the very edges, at the very top of the spikes. And because we elucidated both structures at the same time, we were able to compare how a virus, how St. Louis encephalitis matures from this to this on the right. And we know already, this is very well studied in the literature, that the PR part is cleaved right here by a host enzyme called furin. And the E protein, which is here arranged in a trimer, lands on the virus form dimers. And these dimers are actually bring the infectivity to the virus particle. And as you can see here, we have the glycosylation in E pointing forward as it was here, but in a different configuration. We are able to investigate several aspects, and we are now paying a closer attention to these issues here, or these characteristics, such as the role of histidines, the role of the lipid pocket, which is located right here, and how we can, why we're lacking the data for the capsid protein, which was supposed to be located back down here in the interior of the virus particle, and how this cleavage would result in maturation from a more mechanistic point of view based on high resolution molecular data. So what we have ahead of us, we're going to investigate some of those conserved histidine residues, hoping to draw more insights that will be useful for the entire orthoflavivirus genus. We're using recombinant viruses and evaluating how those viruses perform in mammalian and mosquito cell cultures. We're obviously looking for attenuation. Even though we have dengue vaccines available now, there are so many other flaviviruses, orthoflaviviruses like St. Louis encephalitis are lacking vaccines. And in some instances, especially in the Americans, there are some orthoflavivirus derived diseases that need are in need for a vaccine candidate. And we're also validating that, hopefully, in some in vivo models. So I'm gonna finish my presentation in a couple slides. I would like to thank all the team at the virology group here at CNPM. We have several collaborators from within CNPM and outside CNPM which are assisting us in this. So this is me, this is Samuel and Lais, which are the main authors of this study, and these are our funding agencies here. And finally, this is us saying farewell to you guys over there in Hungary from the building site of Orion. So as you can see, it's a very large building site. Nowadays is pretty much a huge hole filled with mud, but that's something actually positive because Project Orion is under construction and we're just finishing the part in which we lay the foundations. So we expect to have a building rising from the ground in the next semester or so, and have it concluded around at least the construction part around 2028. So I'll leave this with you guys. We have a website. If you reach out to cnpem.br, as in Brazil, there is a website full of information on the Orion project. There are versions in English, and we also have a booklet in Portuguese and in English for all of you who want to know more about the project. And obviously, do not hesitate to reach out if you want to know more about it. It will be my pleasure to make a connection with any of you. So thank you very much.
Thank you, Rafael. Thank you. So, Professor, please try to share again the slide.
Okay. I hope this time it will be all right. And okay, can you see my slide now?
No, we see you, but...
Slide?
Okay, we see your slides. It's not in a full mode, but this is great. So if you can just go with this is great. Yes.
Okay. Okay, ladies and gentlemen, sorry, I'm so sorry that, technology is not supporting me very well. Here is the outline of my presentation. I'll give a little bit out. Approach very organically in Bangladesh and covering all these sectors. And we have been following a new strategy at the moment. And that strategy is based on the experience of our COVID-19, and which we are translating that strategy into One Health programs. And that will be my major issues of my presentation. You all know about these things. One Health started its journey in the beginning of this century, but people were defining it in their own way. And later on, quadripartite, particularly WHO, FAO, One Health, WA, as well as a UNEP, decided to develop a One Health definition with the help of One Health high-level expert panel, and they give a definition which is about. One Health is an integrated, unifying approach that aims sustainably balance and optimize the health of human, animal, and plant and ecosystem. Most importantly, this definition has recognized the interconnectedness of the health of human, animal, wildlife, plants, and wider environment. And it is functioning on the basis of communication, coordination, capacity building, and collaboration among the sectors and breaking the sectors or silos. And with that thing, you all know about that thing. Currently, quadripartite has developed three pathways of change, what they call the theory of change, mostly based on the policy, legislation and financing, and also organizational development and knowledge sharing. And we are aligning our activities in that line after COVID. And but most importantly, Bangladesh is a very typical country for One Health. Why? It is a very small country, only 56,000 square mile area. It is a great delta. But importantly, it is a huge population, 180 million people. It is one of the highest density populated country. And not only the population, it is also known as a hotspot for infectious diseases, emerging infectious diseases. We have avian influenza, Nipah, anthrax, rabies, all are endemic. It faces a lot of outbreak of the vector-borne diseases. It has a burden of foodborne diseases. And of course, we are also facing the challenge of loss of biodiversity and antimicrobial resistance. And with this background, when avian influenza emerged in 2007, we are very nervous because it was a growing industry in the poultry sector. And how can we really deal with these things multi-sectorally? That was our discussion. That time I was the Vice Chancellor of Chittagong Veterinary and Animal Science University. We have taken an initiative, very informal initiative, to bring different sectors together, what we can do in a One Health approach to deal with the avian influenza. And that time also we are having epidemic of Nipah viral infection. So we are having a discussion. Finally, we had a stakeholder discussion, consultation with the UN and other organization, development partner. Finally, you organized a conference in 2008 in Chittagong, and we made a declaration, what we call Chittagong Declaration for One Health in Bangladesh, and also we launched a new civil society platform. And the name of that civil society platform is One Health Bangladesh. And that is how we started our journey in the One Health. And here you can see some of the milestones of our journey. One, as I mentioned, in 2005 to 2007, we are dealing with the diseases like avian influenza, Nipah. In 2008, we organized a conference where we established a One Health Bangladesh. And in 2012, in collaboration with development partners and intergovernmental organization, we developed a One Health First strategic framework, One Health strategic framework for the country. And that was endorsed by three ministries: Ministry of Health, Health and Family Welfare, Ministry of Fisheries and Livestock, and Ministry of Environment and Climate Change and Forest. And after that, we established a One Health Secretariat, and then we also, developed a sort of action plan, and that is how we have been implementing One Health in Bangladesh officially, and One Health Secretariat and One Health Bangladesh were working hand in hand. Now, after the COVID, we revised the One Health strategy framework again, and that is now under function, and that is the third revision of our One Health strategy framework. And this is how the One Health Secretariat works. There are 6 areas that we really focus with the One Health Secretariat. And One Health Secretariat is manned by 3 ministries people, and it is supporting the policies, advocacies, monitoring and evaluation, health capacity building, priority research and coordination development, One Health action, as well as coordination. Those were the key areas of our One Health Secretariat. And what else we do? We do promote a lot of knowledge sharing, and there is a data sharing among ourselves. One Health evidence-based surveillance data dashboard located in One Health Secretariat. Also, AMR data has a dashboard and coming across human and animal and environmental data there. And we organize a lot of high-level discussion as well. One of the most recent one, the 17th December, the high-level consultation on One Health in Bangladesh, opportunities and challenge, under which all 3 ministers, we call advisors, they were there, and secretaries from 5 ministries, 7 director generals of different departments and high official, and they made a strong political commitment to go ahead with the future multi-sectoral alignment and also some action program for One Health. And other than thing, we do a lot of joint outbreak investigation, AMR activities, international collaboration during COVID as well. We have a training program for epidemiology, field epidemiology training program for public health as well as for veterinarians, and joint simulation exercise. And we also did a sort of zoonotic disease prioritization and JE, joint outbreak, joint external evaluation, coffee and star, all those things are being done jointly. And we have strategic planning and resource mobilization, as I mentioned earlier. And also we have developed a One Health investment case studies, action plans for AMR, REVI, size PI, and joint proposal for pandemic fund and pandemic fund multi-country proposal as well. And it has been recognized internationally as well in different publications, including CABI as well as in the Lancet. What is important that we have been developing a partnership with University of Pasay and Chittagong Veterinary University and Russia University to strengthen our research capacity for dealing with the zoonotic disease, in particular focus on the Nipah. That work is undergoing in collaboration with Gabors and other colleagues, and that is one important example with our international collaboration. It is not only with you, there are other international collaboration, including Royal Veterinary College and some other universities in the UK, USA, and we have been partnering with them to undertake some of the activities. And it is not only the infectious disease, we are going beyond that, and in particular, we are focusing on, the pollution, production system in agriculture as well as in animal and ecosystem diseases, in addition to our infectious disease. And this is one very growing concern of environmental ecosystem health management. That is also things going on. We have a very strong community of practice in Bangladesh, and that is going on with their regular conferences. And we develop a sort of system whereby all the partners come there. Every 2 years we organize, and as you can see here, how the number of participants are growing. And the last conference we had in 2023, it was very well attended by our different partners and organizations. One of the beauty of our One Health strategic framework in the coming 5 years, and it has a 5 outcomes, including it is very aligned with the joint plan of action of the quadripartite UN organizations. And that includes One Health institutionalization, pandemic management, One Health systems, as well as healthcare, the antimicrobial resistance, and as well as the environment. It has 11 components, and it covers from the government and coordination to strategic communication, workforce, laboratory system, surveillance system, outbreak control, food safety, and antimicrobial resistance, and ecosystem management. And there is a sort of coordination mechanism from national level to downward, our what we call lowest community level, coordination system. And it has been divided into 6 major areas: National One Health Advisory, which is a very political one, interministerial, intersectoral, and secretariat that I have already mentioned. And it has also in the division, subdivision, and district level coordination. Our current working is a very leadership is rotating among 3 sectors, and it might be included other sectors later on. And there is some good indication of funding. We got the 25 million US dollar project on pandemic funding, which is a very One Health based program we have developed. And we are also having a One Health project towards 2025 million with the support of World Bank. And that is a joint activities in 3 ministries working with One Health activities. And they are mostly focused on the integrated disease surveillance and early warning system, intensive effective national laboratory system, enhanced to the One Health workforce involved with the One Health coordination mechanism. And the One Health project that we have been supported by, by the government, 3 ministries are working together for joint surveillance, laboratory workforce, system strengthening, as well as capacity building. More importantly, cross-border, because Bangladesh is surrounded by different countries, and border is a very important issue, and policy harmonization, regional networking. Those are all these, next 5 years activities that we have planned, and we are having funding from different agencies for that one. And in that way, we are overcoming the financial crisis, as well as we are trying to have the strong institutional foundation based on these things. We do have a challenge as well. It is not without challenge, limited engagement from the policymakers, and that is why we are pursuing very strong advocacy with the policymakers. Complex funding system, it is not straightforward. It is based on the very silos, and we are trying to break that one through this One Health project to cite an example that, yes, we can work together with the different ministries and sectors. Absence of One Health coordination at field level, and this strategy framework, the third one that we have developed, will be focusing mostly to go up to community level, regular policy advocacy, inadequate community awareness. That's what is one other area we are trying to overcome that one, and inadequate utilization of One Health workforce within the government system. Those are the challenges and way forward. This is what, as I said, the One Health new strategic framework, advisory committee, and also for One Health focal point at the ministerial level, One Health coordination committees at sub-national level, sustainable multi-sectoral funding, regular advocacy, community exchange, and as well as workforce development. Those are our way forward for the One Health. And one of the most exciting experience that whatever happened, whenever we convene under the banner of One Health, people come together very easily. This is one important thing. Even during COVID time, when we are facing a lot of problems, the one, human health, animal health work together in the laboratories to detect the cases. So with these things, One Health is going to be one of the priorities for our national program in the next 5 years. That is one thing. Thank you so much, and I'm extremely sorry for having the technical problem. Thank you so much, Gabor and colleagues. Thank you so much for giving me the opportunity to share these things. Thank you.
Thank you, Professor. Thank you. It was great to have you here. Thank you very much.
Thank you very much. Thank you.
So thank you for all. I don't like long speeches, so I have one wish, okay, to meet again in 2030, 2031, depending on the administrative part, but no joking, I was happy to have you here. I hope you enjoyed. I hope you found your new networks, new connections, and I wish you a pleasant journey way home. Thank you very much.